Okay. Hello, everyone. Another Tuesday is upon us. I'm Dr. Garrett Smith, known as the nutrition detective around these here YouTube parts. And today we are going to do an episode on parasites, helminths in the research, often called, and how they thrive in environments that are toxic. They need that, and how we address them permanently versus what everybody else is doing that kind of works for a short period of time, but makes them sicker long term and makes the parasites able to come back. So, for those of you who don't know, the Love Your Liver program and or working with our health facilitators, we have had many reports of people doing the work and then telling us that things that appear to be parasites are coming out in their poop. Key thing here, we are not using herbs or meds or other supplements to try to kill the parasites. We do not try to kill the parasites. We do not put toxins into our system, because if it's, as Kelsey said to me once, she said, if it's toxic to parasites, it's probably toxic to you. If it's toxic to bugs, it's probably toxic to you. We're just bigger, and it takes us longer to accumulate the toxicity. That sounds familiar, right? So when she put it that way, I was kind of like, yeah, so people want to see outright toxic effects from these toxins they're taking. It may not happen outright in you, but as we know here, toxicities of different types accumulate, they synergize. One toxin can cause a problem with another toxin, such as like pesticides slow down your ALDH, your detox enzyme, which then causes you to accumulate more vitamin A toxicity. So the two can be different toxins, but cause each other to accumulate. So what do we work on? We work on removing the toxicities, removing the intake of toxicities or minimizing them. We work on facilitating the removal of stored toxins, and we work on giving the body the nutrients that it needs to detox the toxins and to protect itself from toxins. So, as I was saying, we often have people pass what appear to be parasites. Now, if people say, well, how do you know they were parasites? I'm saying what appear to be to them. This is what they tell me. And then let me give you one recent story. We had a woman who was doing the program, and she was also doing Kelsey's recipe for niacin enemas on a daily basis. She said soon after she started them, she started in the enema, she started passing what appeared to be small worms. Okay. She kept doing them. She came to my office hours because she was a testing and consultation client. She came to the office hours and she said, should I keep doing these? And I said, yeah, it's doing what it's supposed to do. So what does flush niacin do? Kelsey has evidence of, in the research, Kelsey's our niacin expert, our flush niacin expert. We don't use any other form of niacin. We use flush niacin, nicotinic acid. She, where was I going with that? Anyway, so she was passing the parasites. She said every time she did the enema, she would pass more parasites. And I said, just keep going. She did. And what did she say at one point? She said the parasite, whatever it was, stopped coming out. Is flush niacin an antiparasitic? No, Kelsey's got the research evidence on about 400 different enzyme, or some might even say detox pathways that you need flush niacin to run. You need the NAD from flush niacin to run them. So if we start starving the parasites, so what we are doing here, we do not take meds or herbs to kill these things because they are toxic. Think about this. When people say they're having Herxheimer reactions from things dying off, but they're taking toxic things, could it just be that they're taking something toxic and they get side effects and they are convinced they have problems and they took a toxic med and they get side effects and they go, oh, it's definitely parasites. They're really angry now. Oh, they're they're dying off and they're really angry and they're going to attack me and make my brain do certain things. And I'm like, give me a break. There is not like an army of communicating parasites all through your system that is making you go bonkers and give you all these side effects. No, you took something toxic and you're suffering the side effects. Now, as we're going to get into, as I'm going to show you tons of evidence of, parasites are little storage containers for specifically toxic metals. Oh, but vitamin A comes in and copper comes in too. Imagine those two little rascals joining the party. So imagine, let's just say, if you did kill a bunch of parasites and your digestive system, which does what? It digests. If it were to start digesting these parasites that are completely full of toxins, where does it release the toxins into? Oh, your system. And then you absorb them. And then you basically unleashed, as you're going to see, some of these toxic metals are 2,000, almost 3,000 times higher concentration in the parasite than in the host. The host would be you, or the animal that has the parasites, or the fish, or the shark, or any of that stuff. So one of two things is happening when you take antiparasitics and you feel bad. First of all, you're taking something toxic. First, one of the things we always do in this work is if somebody is on a medication and they start getting problems in the program, our first suspicion is always the medication. If somebody's taking an herb that's designed to kill something and they have side effects or they have new symptoms, we always assume it's the herb, the toxin, the toxic thing that they're taking. Okay? We always assume that first. So it could be the med over time. Don't think that just because a med didn't give you side effects for the first 2 years you took it, that they couldn't already all of a sudden start. That's called accumulated toxicity or accumulated damage. Don't think that just because you've taken black walnut 5 times before for parasites, because they keep coming back because you didn't solve the root cause, that the 6th time you take black walnut, it's not going to cause you any problems. Remember, my car worked just fine until it broke. That's things fail all at once. They don't usually fail a little bit at a time. Okay. So now I want to go over the super chat that Houndjog just so happened to start the day with. So Houndjog's super chat was, "Hello, Doc Garrett. After using 4 coffee enemas daily, followed by 2 MMS enemas," and she's also on the Love Your Liver program, "I passed a bird's nest." So imagine what she's describing, right? Bunch of things twisted together. "Was this parasites or the gut lining? Thank you, and you are loved." Well, thank you very much, Houndjog. So we have a couple of things here going on. We have the Love Your Liver program, which is starving the parasites of their toxicity, as you'll see. We have coffee enemas, which induce bile dumping, and then basically bile-induced coffee diarrhea. That's what enemas do, okay? They're inducing diarrhea, and coffee induces a bile dump, and so you have the coffee enema causing the bile dump and then inducing the diarrhea. So it's just basically a short-term bile flush, if you will, okay? And 2 MMS enemas. Okay. What does chlorine dioxide do? It is an oxidizer. Our main detox processes for organic toxins, as organic as in carbon, hydrogen, and oxygen, not like organic food. It means they're not like toxic metals. They're organic toxins. They're made of carbon and hydrogen and oxygen. It's weird that they mix those two terms up. You know what I mean? So MMS is an oxidizer, so it would break down toxins. Like NAD from flush niacin is the gasoline to run your oxidative detox systems. So she did these things, and she's doing the Love Your Liver program, and she passed what looks like a bird's nest of parasites. What was I just explaining to you? So we have a second person live saying that they just passed what very well could be parasites. The lab I knew that used to analyze parasites, like if you were one of those people who would want to collect from the toilet what came out of you and have a lab analyze it and tell you, is this parasites? Well, that lab, that was parasitetesting.com. They have stopped doing that. I do not know why. Maybe people just sent them too many weird things. Oh, and I do want to let you know about parasitetesting.com. The guy there, the main guy there, has specifically stated himself that they have helped fix Morgellons. And you want to know what he found Morgellons to be most closely connected to? The toxicity induced by dental materials. He says that as they get people's dental materials changed out for stuff that is not as toxic to them, Morgellons just happens to go away. We see the same patterns all over the place. You fix toxicity, the bad things don't grow on you, they don't live in you, they starve and they die. So that was pretty cool. Thank you for posting that, Hound Jog. That was great. A great example of what we're doing. Okay, she said 5 activated drops. Yes. So let's get into the research. First, let me just show you. Joe's going to post the link in the chat. This is just the research link. So all the research links we're going to go over today, they are all arranged so nicely for you by moi. So if you want to look these up, you can. They're all right here. So there's that. Otherwise, so I don't have to type them all into the into the chat or copy and paste them. So first, I want to go over an idea here. Some people are saying, there's 2 things going on here. Some people are saying that parasites, we don't have enough parasites, and parasites help cure diseases. And so you need parasites. And I even think some of the same accounts on Twitter are saying parasites cure diseases and parasites are the cause of all diseases or cancer and all. Okay. I want to understand to you that the way parasites interact with toxicity explains both. Okay. Let's start with a simple idea. If parasites, as the word is going to be used as we go, if parasites are actually what they call sinks or little depots, or to translate depot, little deposit areas or storage containers for toxicity, and they actually relieve some of the toxicity of the host by pulling the toxins into them, then they could actually benefit health in certain conditions. Okay? It's not that they're stimulating your immune system. What if they're just taking away toxicity? They're just holding on to it away from the system. You understand? So then they say parasites cause cancer. Well, just think about this. Let's say you have a bunch of parasites, and as you'll find in the research, as we're going to go over, parasites very quickly soak up a ton of toxicity. Okay? But then they hit their limit. So then what if you kept taking in the toxin and the parasite vault is full? Well, then what's your body going to do to store more toxicity? Well, it's going to make more tissue, especially fatty tissue. As you're going to see as we go over this, it's going to make fatty tissue like tumors, and tumors are specifically made to hold toxic tissue. To hold toxins in them. So imagine between the parasites, if they don't hold enough, then you got to start making tissue. And then people later go, parasites cause cancer. Well, maybe the parasites came first because that was the first line of defense and you overwhelmed them. And then you see cancer. And then then people look at it and they go, cancer caused, or the parasites caused the cancer. No, the toxicity caused the cancer. So there's going to be different toxins that feed different parasites. There's going to be different toxins that obviously feed different cancers. But you have to understand this. So can parasites help diseases? Yes, by soaking up toxicity. Can parasites be associated with other diseases of toxicity? Yes, that's an overwhelming amount of toxicity that the parasite sink couldn't hold enough. This all makes sense when you understand what they're doing. So then, imagine if all of a sudden you try to go and kill and destroy all of your parasites. What are you potentially dumping back into your system? The toxins they stored. Let's say you poop them out. Okay. Well, did you fix the condition in your body that encouraged them to grow? No. Did you probably actually get rid of all the parasites? Probably not. So, it's like they say, if you had 2 humans, you could repopulate the world. Well, so how many parasites does it take to repopulate your gut? If 1 escapes or 2, can they repopulate your gut? Or if there's still some eggs left, can they repopulate? Yes. Did you fix the root cause of what they fed on and thrived on? No. Why do you think people's parasites keep coming back? And then if you're exposed to this is one thing that has to happen. You have to be exposed to them for them to come in. They are not like terrain theory people. You're not going to have parasites like, what's the term? They're not going to change from a virus to a bacteria to a yeast to a parasite. That's not happening. Parasites are actually, like, you can visualize them in real life, like they are real. They are little creatures. Okay. They don't appear magically from what's pleomorphic. You're not creating these organisms pleomorphically. Okay. These are things that get into your system. And if you have the right conditions, if you have the conditions for them to feed and thrive, they grow. Does everyone have these conditions? No. That's what you're going to see later on in some of the animal studies where they say, oh, only 1/3 of these animals had parasites, but they all lived in the same forest. Or only 60% of the rats had parasites, but they're all urban rats living in the same like sewers. So why don't they all have it? Why don't all humans have all the parasites all the time? Because not everyone is susceptible to them. Not everyone has the toxic chemical soup that they love, that they feed on, and not everyone's exposed to them. So moving on. So now we'll go to that one, Joe. Just want to show you some of the evidence of where people will say, parasites fix disease. So here we have Helminths. Just think of Helminths as another word for parasites, and immunological tolerance. Okay. This paper is mainly, they're talking about how to make people not reject organ transplants. Okay. Helminth parasites may provide novel strategies towards achieving this goal. Helminths are remarkably successful parasites. They currently infect more than 1/4 of the world's population. Mostly places with poor sanitation and poverty. Keep that in mind. Do we ever talk about people who don't eat enough meat and zinc and these other things that go along with meat, and how poverty is really, people's diseases and poverty is really reflecting their lack of eating meat? Okay. It is now well established that the parasite's success is the result of active immunomodulation of their host's immune response. We're going to go over chicken or egg here. Although this primarily secures ongoing survival of the parasites, Helminth-induced immunomodulation can also have a number of benefits for the host. Significant reductions in the prevalence of allergy and autoimmune conditions among Helminth-infected populations are well recognized, and there is now a significant body of evidence to suggest that harmful immune responses to alloantigens may be abrogated as well. I'm not going to look it up right now, but I'm assuming that this means the transplant. Harmful immune responses to things of that nature. So we have been over here. We have been over this here before. Allergies are vitamin A toxicity. Go watch the anaphylaxis show. Allergies are vitamin A toxicity. Autoimmunity at its root is vitamin A toxicity. How do we fix both of these? Well, we see it happen. So imagine if the parasites come in, they find a person who is vitamin A toxic, and they get infected. especially in areas of poverty where they don't get enough meat and zinc, then the parasites feed on the vitamin A. It helps keep them alive. That's what they need. And you're going to see this in a bunch of parasites later. So they're saying it helps allergy and autoimmune. We talk about fixing allergy and autoimmune by having people avoid vitamin A. So do you see how you can have both? So let's keep going. Okay. Extraintestinal Helminth infection reduces the development of colitis-associated tumorigenesis. Okay, so Joe posted a definition to alloantigens. Alloantigens are antigens present in some but not all individuals of the same species, and they can trigger an immune response in individuals who lack them. These antigens are often involved in transplantation and transfusion reactions. Okay. But let's go. So extraintestinal, this means outside of the intestines. So parasite infections outside of the intestines reduce the development of cancer that's associated with colitis. Okay. Colitis-associated colorectal cancer is one of the most common cancers and is closely related to chronic or deregulated inflammation. Let me break that down for you. Chronic or deregulated inflammation, toxicities and deficiencies. Okay, if somebody's got colitis, do you think their bile is less toxic or more toxic? More toxic. It's actually eating away at their guts. What is cancer? Toxicity accumulation. So if you have tons of toxic bile going through your guts all the time, where might your body want to start absorbing it, soaking it up first? In your intestines. What is cancer? Toxicity storage, okay? We have analyzed the role of Tinea crassiceps-induced anti-inflammatory response in determining the outcome of colitis-associated colorectal cancer, CAC. We show that extraintestinal crassiceps infection in CAC mice inhibited colonic inflammatory responses and tumor formation and prevented goblet cell loss. The parasite is soaking up toxicity, so they get less, they don't have to form the cancer. If you continued to give these mice the same toxin and you overwhelm the parasites, what do you think is going to happen? The cancer. Okay. Thus, immunomodulatory activities induced by Helminth infections, so the immune system or immune system changing activities induced by Helminth infections may have a role in the progression of colitis-associated colorectal cancer. Okay. Okay, so what is your immune system? This is a very important early definition. Your immune system is your cleanup crew. Stop thinking of it so much as an army. It's not a defensive army so much. It is a cleanup crew. So if you had too much waste all the time, not only are you poisoning your own immune system, but you're overworking them. So you're overworking them and you're poisoning them. So you see over time, your immune system goes down. Your immune system is getting poisoned too. Your bone marrow is getting poisoned. Your liver is getting poisoned. All your other glands and organs that are associated with your immune system are all getting poisoned. So what you see, just so you know, what you see usually in acute infection, acute toxicity is you see the immune system go up because it can still respond. If you dumped a bunch of garbage into your system, what should happen at the start? Your cleanup crew should increase its activities. So you see white blood cells go up in acute infections, which are really acute toxicities. Then over time, the poisoning lowers your white blood cells. And then they say you have a low immune system. And what I can tell you is that we help people fix low immune systems over time. And the funny thing is we go right backwards through the same progression. They're low, they start getting higher, and then they might even go a little high before they come back to normal. Because wouldn't it make sense if you were cleaning out stuff and all of a sudden your immune system starts to say, well, wait, we can make more guys now to help, and we're getting the winning hand, the upper hand on the toxicity. Now we can clean it out. So what are the parasites doing for all of this? The parasites are soaking up toxicity. They're doing the job of the immune system. So the immune system has less toxic load so it can do more of its job. Does that make sense? Okay. When parasites are good for health, cestode parasitism increases resistance to arsenic in brine shrimps. I'm going to tell you guys, a lot of people may watch this video later and go, well, he doesn't have a lot of human research. They're not doing a lot of human research. Why do you think that might be? Because they don't want you to know that you having parasites means you are toxic. They can't tell you that. So they're going to do a ton of research in fish. Tons of, I see Mike Roberto. So if you guys don't know who Mike Roberto is, well, Will's there too. And thanks for coming, Will. But Will is Will of Europa. Will, you can put your Twitter handle on there. Will posts good stuff on Twitter too. And then Mike Roberto, MicroRoberto at Mike Roberto on Twitter. He posts awesome research, just destroying vitamin A on a regular basis. So I highly suggest following both of them on Twitter. And Mike said, finally made it to a live stream. Thanks for everything you do. This is a fascinating topic. Love how it fits into toxic bile paradigm so well. Thank you, Mike. I appreciate that. One of the things that really makes me feel good is how many like really, truly intelligent people that have found this work, and then they look at it and they put it into their brains and they go, this makes so much sense. I'm like giving myself chills with this idea, because that's actually the biggest compliment to me, is when I have smart people telling me this makes so much sense and this answers so many questions. And I just, like I said, I get the chills. So anyway, thank you, gentlemen and ladies too. I mean, Kelsey's one of the smartest people I've come across, and she has helped the program so much. So yes, it's all sides. Okay. Pay attention. Here we show for the first time that parasites can increase host resistance to metalloid arsenic, focusing on a clonal population of brine shrimp from the contaminated Odile and Tinto estuary in southwest Spain. We studied the effect of cestodes on the response of Artemisia to arsenic. Let me make this a little bigger for you guys. Acute toxicity tests, the 24-hour lowest concentration to kill 50% of them, and found that infection with parasites consistently reduced mortality across a range of arsenic concentrations. Parasite infection consistently reduced the risk of death across a range of arsenic concentrations. You're not trying to fight here's the mind blow. You're trying to reduce the toxicity that feeds the parasites. You're not trying to just kill the parasites. Killing the parasites doesn't fix the problem. It may actually unleash the problem more into yourself. Okay? And then you're taking toxic things to do it. So it's like a double whammy. You're not fixing the root cause. You're taking something toxic, and you may be unleashing the toxicity that the parasites were holding. Also, people can be like, can I take this ivermectin or this other herb or whatever to kill the parasites and then do the LYL program? Why? Why are you going to try to make yourself more toxic before you go and try to detox? Why are you going to go and damage your detox systems before you try to detox? Like with arsenic, if you haven't seen my arsenic and rice preparation video, boy, if you're not preparing your rice right, you're going to be taking an arsenic. So fix that. Like, don't put the poison in that would feed the parasites. Then not only are you healthier, but you don't need parasites to help you out. So they're saying here, infected individuals like brine shrimp showed higher levels of catalase and glutathione reductase activity. This is your body fighting against them. It's still fighting. So does the brine shrimp body or your body, do you love the parasites? No. It's more work for your system. If your body can compensate, we can have less risk of death if you have parasites. But here I'm going to tell you, if you have arsenic toxicity with no parasites, are you still unwell? Yeah. If you have arsenic toxicity with parasites, but you have a little lower risk of dying, are you still unwell? Yes. Neither of these situations is good. Okay, moreover, infected Artemia had a higher number of carotenoid-rich lipid droplets. The infected shrimp had a higher number of carotenoid-rich lipid droplets. And of course, they're going to justify it and say it may protect the host. No, they're just accumulating all sorts of toxicity. It's going to be a problem eventually. Poisons of a feather flock together. Oh, we got arsenic and carotenoids. Great. What if the parasites like the vitamin A? If you don't know, carotenoid is what we call plant vitamin A. What if the parasites like this too, and that's why the parasite ones have more in them? Some of these parasites will eat certain toxins, feed on them, and some of them will avoid certain toxins, but they can still affect your detox, okay? The vitamin A connection will make more sense when I get to the whole vitamin A section. I think the 5 or 6 papers that I have on vitamin A and parasites. Okay. So now I'm going to try to run through these fish ones quick. So again, like I said, there's lots of fish studies, but then you get to humans and mammals and there's very few. I'm sure that wasn't planned. Biomonitoring of heavy metal pollution using Acanthocephalans parasite in ecosystem. An updated overview. I'm just going to start saying acantho for this just because this one comes up a lot. This review updates and focuses the light on the relation between the toxicity of heavy metals resulting from bioaccumulation. Gosh, who talks about that? In fish, and the parasite bioindication role and its infestation. Now, let me see. I think this is the one where we get to connect bile to parasites. Here we go. Let me get that out of the way. There we go. Interestingly, it has been reported that the concentration of heavy metals was higher in the intestinal parasites, acantho, several thousand times higher in comparison to the fish tissues, as in the liver. Thousands times higher than the liver, kidneys, gills, and muscles. The concentration of toxic metals in the parasites was several thousand times higher than the liver. Do you understand that these parasites are helping the creature to not be so toxic? This next part's important. The presence of the bile acids in the lumen of fish, so that's like the inside of the gut, resulted in the formation of organic metallic complexes, which are easily absorbed by the worms due to lipophilicity. Felicity is loving fats. Wait, I just want to make sure. So bile helps you absorb fats. The bile acids formed organic metallic complexes with the heavy metals, which were then easily absorbed by the worms due to lipophilicity. Are you understanding what this just meant? It means that you folks on high-fat diets, you absorb more fat-soluble toxins, organic toxins from it. Wait, organic metallic complexes. Wait, you absorb more fat-soluble organic toxins like I went over in my weight loss and toxicity video. You store those in your fat, and you're absorbing more toxic metals. A high-fat diet causes you to absorb more toxic metals. That's what they're saying here. Why were the intestinal parasites higher? Where does your bile go first? Into your gut. Malik et al. Concluded that the concentration of lead and cadmium in Cestoda Paraotigmatobothrium species and Anthrobothrium species were higher than their levels in kidneys, liver, kidneys, and gonads of shark collected from Iranian coastal water. So these parasites are concentrating lead and cadmium too. This finding has strongly supported the theory that the Helminths parasites are extremely sensitive bioindicators that may serve as an early warning, particularly in the sensitive low-level environmental threats. Therefore, they may also have beneficial effects on the health of their host by acting as heavy metal filters. If you had a filter that was filtering tap water, collecting all those nasties in it, And then you decided, hey, let's just take that filter and grind it up and put it in the water we're going to drink. That would be like you taking parasite medications or parasite herbs. Let's just break up all the crap that's in there and just put it back into the system. I'm sure it'll be fine. So let me see. I think that was it. Yep, that was all for that one. So some of these other studies I will have in here. This was another bile one. Parasite responses to pollution. What we know and where we go in environmental parasitology. Gosh, what is that connected to? Toxins in your environment or the animal's environment? The study of parasites and toxins in the environment. Over the last 20 years, 3 main research directions have been shown to be highly promising and relevant, namely parasites as accumulation indicators for selected pollutants. Parasites thrive where there is toxicity. Parasites thrive so much where there's toxicity, they use them to measure the toxicity in the area. Parasites as effect indicators and the role of parasites interacting with established bioindicators. That's like, so blood levels, blood markers, labs. Okay. I think I need to jump to the next part. Okay. Laboratory studies on the accumulation of lead suggest that acantho take up the metal in the form of bile metal complexes. Remember, for those of you out there who still think fat is good for your bile, first of all, fat detoxes nothing. No fats detox you. Fats are a toxic load on your liver. Okay, you eat, then people think, I'm going to eat a bunch of fat to dump a bunch of bile because Dr. Smith says bile's the problem, so I'm going to eat a bunch of fat to dump it. Why is this not a good idea? Because bile is needed to absorb fat. Fat is absorbed via bile. They bind together, and then you absorb them. So the more bile you dump with the more fat you eat, the more of it is reabsorbed. Does that make sense? They bind together. That's how they help you absorb it. And it seems that bile likes to bind to metals too. When exposed to metals, organometallic complexes are formed in the liver of many vertebrate species, which then pass down the bile duct into the small intestine, where they can either be reabsorbed by the intestinal wall, especially if you eat a lot of fat, and run through the hepatic intestinal cycle. This is hepatoenteric circulation. That's another word for it. Or they can be excreted with the feces. So you, all muscle meat carnivores who poop like once a week or once every 5 days, or some of you guys once every 2 weeks, I don't know. You're not getting rid of the bile. You're reabsorbing everything in that poop multiple times. If organisms are infected with acantho, the parasites interrupt the hepatoenteric circulation of metals. They interrupt the cycling of that, as they were shown to rely on the uptake of bile acids from their host's intestines. The parasites rely on bile acids. Gosh, what's in bile? Toxins like metals? In principle. So what if you made your bile less toxic? So the toxic bile is coming into your intestines. It doesn't feed the parasite because the food is not in it anymore. In principle, all substances entering acanthocephalans and cestodes have to pass through their tegument. Hold on, let me just make sure everybody knows what that means. A natural covering and protection of the body or part of it. So they're absorbing it through their skin, if you will, right? They don't have skin, but they're absorbing it through their, whatever you want to call their skin. Okay, the tegument. You can call it the tegument. Where was it? Is this the one I wanted? Yeah, there it is. Accordingly, if substances can be detected in cestodes and acanthocephalans, they are biologically available in the sense that they are able to cross biological membranes. Oh, I didn't share this one. There they are. Okay. I love the bioavailable copper people, because they're just like copper in your shower water is absorbed into your system. It's bioavailable. Mercury in your fillings is absorbed into the system, it's bioavailable. If a toxin gets into your system, it's bioavailable. So saying bioavailable copper, actually, just so you know, I've looked up the only 2 papers I can find that use the terms bioavailable copper in the actual scientific literature are talking about copper toxicity from bioavailable copper. And if you wanted to know what they're trying to make copper bioavailable with, they're binding it to nicotinic acid. That's the mitosynergy. They're binding copper to nicotinic acid to try to get it in the system with less problems. They're trying to prevent the copper from causing problems by binding niacin to it. Bioavailable copper is horse shit. How's that? It's still toxic. So let's keep going. Environmental parasitology, intestinal Helminth parasites of the cyganid fish Cyganus rivulatus as bioindicators, things that they can keep track of for trace metal pollution in the Red Sea. You're just going to find this over and over, all over the world. Cadmium concentrations in the intestines, liver, and muscle of non-infected and infected fishes were much lower than those of lead, and both metal concentrations decreased in the order liver to intestines to muscle. Liver's highest in lead. Gosh, imagine that. Does the liver accumulate more toxins than anything else? Generally, yes, but parasites beat it. Again, the liver stores toxins. Anybody who says otherwise should be ignored on probably pretty much everything else. Cadmium and lead, I want to pay attention here. Cadmium and lead concentrations in the tissues of fishes infected with G. volubilis or P. elatensis were slightly lower than those in non-infected ones. While in the tissues of fishes infected with SC rubrimaris, they were much lower. The parasites are taking some of the toxic load. They feed on the toxicity. They can actually make you less toxic. That's how we saw in the early thing. They can help some conditions for a while until you out toxin intake them. Okay? By contrast, high concentrations of cadmium and lead in S. rubrimaris are associated with its high ability to accumulate trace metals. Let's keep going. Heavy metals, parasitologic, and oxidative stress biomarker investigations in Heterodotus niloticus from Lekki Lagoon, Lagos, Nigeria. Occurrence of parasites in fish could bioaccumulate heavy metals by as much as 200% more than values present in fish tissues. This is what they're doing. They're grabbing onto toxins. Okay. So I want you to remember, all these fish are swimming in the same water. So they should all be exposed to the same parasites. Why do only 70 out of 100 fish sampled were infected with the parasite? I mean, seriously, when you listen to these people who say, everyone has parasites, like these fish are in the same lagoon and only 70 out of 100 have the parasites. So do you think every human walking around has parasites? No, they don't. Stop with the nonsense. You'll see the Fox study, like I think it was something like 20 out of 80 had parasites. They all live in the same forest. They're all eating the same like dead animals and hunting the same things, and they're exposed to the same toxins and the same pathogens, if you believe in that. But not all of them have parasites. So T. niloticus bioaccumulated cadmium, nickel, and lead between 65 to 100 times more than the liver and 12 to 200 times more than the intestine. Other metals bioaccumulated from host tissues by the parasite had the magnitude between 1 to 12 times as the liver and 1 to 30 times as the intestine. There were significant differences in the activities of antioxidant enzymes. They're mainly talking about glutathione between the parasitized and non-parasitized fishes. Fish tissues also showed histological alterations ranging from mild infiltration of inflammatory cells. That's like white blood cells going in there because they're trying to get the toxins out. Imagine that. Infiltration of inflammatory cells. What if we put it in a good way? It's your white blood cells going into the tissues to try to remove the poisons. To moderate inflammation and hemorrhagic, as in bleeding lesions. 1 to 12 times, 1 to 30 times, 12 to 200 times, 65 to 100 times. The parasites need the toxicity to survive. Cadmium concentrations in 2 adult acanthocephalans, I'm just not even going to say these, as compared with their fish hosts, and cadmium and lead levels in the larvae of A. lucii as compared with their crustacean host. We're going to ignore the crustacean, but really you don't want to be eating those. Don't eat shellfish. Again, adult acantho, the parasites showed several times more cadmium than did the tissues of their fish host. According to the present results, it appears that the heavy metals cadmium and lead are predominantly accumulated by the adult acanthocephalans inside the fish gut. Where does the bile go? Gut. Bile forms complexes with the metals, and then you reabsorb it. Okay, another acantho study with the uptake and accumulation of lead in this fish host chub. This is a good one. Uninfected chub, as I believe the chub are just little fish. Our fish experimentally infected with acanthocephalan parasite P. levis were exposed to 210 lead for up to 30 days, and I believe this is radioactively marked or somehow marked, and the uptake and distribution of lead within different fish organs, and the parasites were determined at various time points. Highest metal concentrations were detected in the acantho, followed then by intestine, bile. Intestine, where does the bile go first? Oh, into the intestine. Then where does it go? Into the, it is the bile. Then the liver. Wow, what a trio there. Toxic bile paradigm. Anyway, gill and muscle of the fish host. The muscle is the least. This is why if you're going to eat meat, you only eat muscle meat. Muscle meat is always the least toxic tissue of the animal to eat. Infected chub had significantly lowered lead levels than in the gills on day 17, in the bile on day 24, and in the gills as well as in the intestine on day 38 compared with uninfected fish. Infected chub, I think these are little fish, had less lead in the gills, in the bile, and in the gills as well as the intestine compared with uninfected fish. The parasites are protecting you from the toxicity. The parasites are there because of the toxicity. The parasites in their own way are protecting you from the toxicity. So how do we get rid of them and get you healthier? We get rid of the toxicity. A subsequent polynomial regression revealed that lead levels for the infected fish ranged below the levels determined for uninfected fish during most of the exposure period. This is the first proof that P. levis reduces lead levels in the bile. Wow, guess bile's toxic, huh? Thereby diminishing or even impeding the hepatic intestinal cycling of lead, the enterohepatic circulation of lead, where the lead is put into your bile, it goes into your guts in the bile, it's reabsorbed through your guts into the portal vein system and goes right back to your liver. So it didn't escape. You have to poop to get rid of the bile. That's the best way to get rid of the bile. So if you're not pooping, if you're not taking things that grab onto the bile, like soluble fiber, charcoal, other binders that we use, you're not going to get it out of you. You might end up like somebody else who has tons of high copper in their system and never gets it out, which may reduce the amount of metals available for the fish organs. This is especially important for ecotoxicological research. For example, organisms used as accumulation indicators may erroneously dictate low levels of pollution if they are infected with parasites, which alter their pollutant uptake mechanism. So what they're saying is, if they analyze the fish in an area, they might mistakenly look like they're not as toxic, the ones that are infected by parasites. So if you analyze a fish that's infected with parasites, you might get a lower lead level than you should, because the parasites helped lower it in them. So they're saying you should monitor the parasites. Comparison of the metal accumulation capacity between the acantho. So again, just another acantho. Let me see if there's anything new on this. Acantho shows the highest accumulation rates. Obviously, we saw like 2,700 times, or have we gotten to that one yet? We haven't gotten to that one yet. Developmental stage, larva or adult, as well as parasite location in the host are suggested to be decisive factors for metal bioconcentration in parasites. As we've already been going over, where would you guess the parasites would accumulate the most toxicity? I'm going to say liver, bile, intestines. That's where the parasites would be to accumulate the most toxicity. Why? Because the liver's full of toxicity. It puts it into the bile. That's toxic. And then that goes into the intestines. 9 elements. So let's look at what they looked at. Arsenic, cadmium, cobalt, copper, iron, manganese, lead, selenium, tin, vanadium, and zinc. So there are some good ones in there. 9 elements were detected in significantly higher levels in the parasites compared to the host tissues. The element composition among parasites were found to be strongly dependent on parasite taxa developmental stage, so whether they were larva or the full parasite, and localization, or where they were located within the host. Intestinal acanthocephalans accumulated mainly toxic elements, arsenic, cadmium, lead, whereas the intraperitoneal, this is inside the abdomen, inside the peritoneum, the nematodes bioconcentrated essential elements, cobalt, copper, iron, selenium, zinc. Well, that's not essential. Go look up cobalt toxicity. Copper, we talk about that all the time. Do we want more of that? No. Lead is a big problem once it gets out of control. And then selenium and zinc. So these things, you can take away good nutrients. So this is why the parasites can cause help. They could also cause problems. Zinc and copper are generally going to often be absorbed together by these things because they're so close in the periodic table. That's the way it works. Oh, but that's the other thing. So if we're talking about places where they don't have good nutrition and then they start getting parasites, what are they not eating? They're not eating meat. So they start losing their zinc or their selenium or both, and they're screwed. And even if they're not eating meat and they're getting their iron stolen away from them, then that can cause a problem. This is why you have to understand parasites can cause problems or they could help, but it's all related to toxicities and deficiencies. Oh, I stopped sharing that one. There's that one again. Let's go back to this one. Okay. I think this is another acantho here. I'll try to hit this one quick. Let me see if there was anything else in here. Oh, here we go. Just to show you how high it can get. Chub experimentally infected with the acanthoparasite P. laevis were exposed to 0.1 milligram lead over 5 weeks. Lead was rapidly accumulated in the intestinal worms, reaching a steady state concentration after 4 weeks. So they have a limit. Gosh, what do you think happens after the parasites are full? What happens to that lead? Maybe your body makes cancer? This concentration was significantly greater than in the host muscle, liver, and intestine, and more than 9,000 times higher than the exposure concentration. 9,000 times higher than this. Lead accumulation in P. laevis increased at higher exposure concentration, but was not correlated with either parasite intensity or with pooled or individual worm weight. So the lead accumulation, as they gave them more, it increased, but the number of parasites didn't change it. I believe they means parasite intensity by the number of parasites or with how big the parasites were. The highest lead concentrations were recorded in those specimens of P. laevis that were attached in the posterior intestine. Interestingly, parasitized chub accumulated less lead in their own tissues than uninfected conspecifics. That means the same species. The ones with parasites accumulated less lead. Over and over, we see this. Another fish. Use of fish parasites as bioindicators. Why do they keep using them? Why are they not using human parasites as toxic metal indicators? Because you'd see behind the curtain. You'd realize that parasites are toxicity, and then you might get to the point where you're going cancer is toxicity, and you realize treating these things with toxins is a bad idea. Free-living invertebrates, notably bivalve mollusks, shellfish, are commonly employed in this role as sentinel organisms. The guard, the watchman. Sentinel organisms to monitor the concentrations of bioavailable metals. Bioavailable can be absorbed. Metals can be good or bad. Good or toxic metals in aquatic ecosystems. Do you know why we don't eat shellfish? They have research where they use shellfish as the accumulator, how to track how much toxicity is in it. Oh gosh, what if there's something even better to monitor than shellfish? Don't eat shellfish. Also, certain parasites, particularly internal acantho of fish, can accumulate heavy metals to concentrations of orders magnitude higher than those in the host tissues or the environment. The comparison of metal accumulation capacities between acantho and established free-living sentinel organisms, so the mollusks, the shellfish, revealed significantly higher concentrations of several elements in acantho than in the zebra mussel, which is commonly used bioindicating organism in Europe. So they're saying that shellfish accumulate toxic metals so well that they were used to study it. But now they're saying parasites are better. That's pretty bad. These investigations provide evidence that the extremely high metal concentrations in intestinal acantho of fish are not the result of a slow process of accumulation, but instead a relatively rapid uptake to a steady state level. They're not just passively absorbing these toxic metals. They are sucking them up. Uptake. They want them. Rapid uptake. They live on this stuff. That's what they do. They can only take in so much, though. Parasites as accumulation indicators of heavy metal pollution. I don't need this one. This one's just kind of... I just liked this word. Conspicuous metal accumulation by parasites. That's what I really liked about this. And orders of magnitude. So certain parasites can accumulate heavy metals at concentrations that are orders of magnitude higher than that in the host tissue. So if you know what an order of magnitude is, it's a factor of 10. So 1 order of magnitude would be 10. 2 orders of magnitude would be 100. So they're saying it's at, hundreds of times higher than in the host tissues. Accumulation of heavy metals by intestinal helminths in fish. Let me see if there's anything interesting in this one. Among these parasites, cestodes and acantho in particular have an enormous, when scientists use the word enormous, it means something big. Heavy metal accumulation capacity exceeding that of established free-living sentinels like the shellfish. Metal concentrations several thousand times higher in acantho than in host tissues were described from field and laboratory studies. Okay, let me see if there's anything else in here. Oh, just again, the mechanism which enable acanthocephalans to take up metals from the intestinal lumen, the inside of your intestine of the host, appears to be based on the presence of bile acids. Again. Continuing. I just want to get to the shark ones. I'm done with the fish. Let me just see. Let me see if there's anything interesting here. We have heavy metal concentrations in adult acantho and cestodes compared to their fish hosts and established free-living bioindicators. We're going to get to more than the fish. Don't worry. Oh, here it was. The lead level of P. laevis was up to 2,700 times higher, and the cadmium concentration was approximately 500 times higher than the mussel of chub, its host. Nonetheless, M. wagoneri contained 75 times more lead and 43 times more cadmium than the mussel of its host, the tench. So people might say, but again, we don't know what human parasites have. I'm going to tell you that to be on the safe side, you better assume that, so terrestrial or land living mammals who aren't, here's why we don't eat fish and shellfish. Whatever is toxic in the water is what is your fish and your shellfish are marinated in. It's what they are breathing in to live. They are sucking it in all day long. There's a saying about like water, buy a filter or be a filter. So what do you think the fish are doing? Do they have filters? No. So they are the filter. So then they get exposed to a parasite and the parasites come in and they're like, cool, heavy metal party. Let's eat it. If the oceans and the lakes were clean, fish would be different. Shellfish just don't eat them. Fish would be different if the water was clean. If you're going to take a bet and say that you think the lake by you is clean, good luck. FAFO, good luck. Lakes are going to hold on to toxicity more because they don't actively flow and they're not as big as the ocean. Okay. Oh, this was the Nigeria one. Did I already go over this one? I don't remember. Oh, this is another one just showing that they're not all infected. I think we went over this one. Yeah, we did that one. Joe, you can delete the extra one in the notes. So here we go. Now we're going to get into sharks, right? Because sharks are apex predators. So what are they going to concentrate? They're going to bioaccumulate all the toxicity below them. Parasites as heavy metal bioindicators in the shark parasite from the Persian Gulf. So now we're just going to show different animals. They measured lead and cadmium concentrations in the liver, intestine, muscle, and gonad of the shark and its parasites, these two. The results revealed that lead and cadmium concentrations in both parasite species were many times higher than in the host tissues. These results strongly support the view that Helminth parasites are extremely sensitive early warning bioindicators. Parasites are extremely sensitive early warning bioindicators. So why do you think they want you to kill them? And they want to tell you that the parasites cause all your problems because they desperately do not want you to look into toxicity. Because they allowed that or encouraged it or paid to have it happen. So parasites are extremely sensitive early warning bioindicators, particularly in sensitive environments under threat, but where pollution levels are presently low. So even where they're presently low, they accumulate it. They also have beneficial effect on also have a beneficial effect on the health of their hosts by acting as heavy metal filters. Again, they are a filter. Your kidneys are a filter. Your liver is a filter. The parasites are an extra filter. Do you want to have an extra filter? No, you don't want to need it because that means you've got toxic metals in you that are supporting parasites. Another one, shark parasites as bioindicators of metals in 2 South African embayments. Again, the parasite in the shark showed accumulation of arsenic, manganese, lead, titanium, and zinc. You're gonna absorb some zinc because it's gonna be in there. That's why I tell you, like, your liver is either gonna be full of zinc or it's gonna be full of copper or some mix of the two. Your liver is going to fill up with either copper or zinc. You don't want it to fill up with copper. That's a disaster, and that's all over the research. And what happens to people in poverty countries where they don't eat meat? They get tons of diseases of vitamin A and copper because they don't have any zinc, and you're gonna fill up with something. These results were compared to baseline values showed that the accumulation of the metals in G plana are orders 100 times plus of magnitude higher than those in the surrounding environment and 2 to 6 times the concentrations of the surrounding host tissues. Okay, let's get into mammals. So what I'm doing here is I'm establishing patterns. We showed it in fish. We showed it in sharks. What did we just show it in? We did sharks. Now we're going into mammals. Heavy metal bioabsorption capacity of intestinal helminths in urban rats. So these are rats. These are in the city rats. Know what I'm saying? We compared the concentration of cadmium and chromium in urban rats, and then they're harboring helminths. M. maniliformis, H. diminuta, and larval stage of T. teneiformis, Cystericus fasciolaris. A higher concentration of heavy metals were revealed in the parasites than in the host tissues. Bioconcentration factor. For both cadmium and chromium absorption, chromium is a toxic metal. Again, chromium is a toxic metal. Do not supplement it. Were more than 10-fold higher in M. maniliformis than in the 3 compared host tissues, more than an order of magnitude. The bioconcentration factor of cadmium in M. maniliformis compared to the liver, kidney, and muscle of the host was 9.116 times higher 14.14, and 17.09 respectively. So again, I'm guessing this means there was the least cadmium in the muscle, just like we say, and there was the most concentration in the liver. Because it's always a pattern that the muscle has the least toxicity. Bioaccumulation factor in chromium of the same parasite in the same host tissues range from 10, 7, and 4.6. High level of absorption in H. diminuta was significantly likewise. The individual bioconcentration factor of cadmium and chromium in H. diminuta compared to the liver, kidney, and muscle of the host was 4.95, 5.94, and 4.67 versus 2.67, 11.56, and 5.59. The mean average concentration of cadmium and chromium in C. fasciolaris was also significantly higher than that of the rat livers. This study claims that parasites of terrestrial land-living animals exposed to heavy metals can be more accurate indicators than the host tissues as new environmental monitoring agents. Next one, significance of intestinal helminth infection and animal sex for mercury concentrations in 2 rodent species. So we got rats. Now we're going into other rodents. We compared the effects of animal gender, species, and intestinal helminth burden on mercury concentrations in rodents. So a new toxic metal. Total mercury concentrations were determined in the liver and kidney tissues of 80 small rodents and 36 bank voles. So these are two different, they're mice and voles. So just remember glariolus. I remembered glarivolus. So voles, glarivolus. So you can keep track of it. Captured in the Ore Mountains. So these are wild living rodents. These are not city rodents. Overall, 25 of the 80, 32% of animals were infected by intestinal parasites. Why weren't all of them? Again, why weren't all of them? The differences in mercury concentration between rodents infected and not infected with intestinal helminths was not statistically significant. This is going to be significant here. Statistically significant difference in mercury concentrations were found only between voles and mice, so the two different species that were not infected with intestinal helminths. So they weren't infected, and they found differences between the species. Okay, this suggests that the differences may be associated with host genetics. Different people, let's even say different races of people are going to detox different. For example, 50% of the Asian population genetics is deficient in ALDH. Genetics plays a role in detox. This might be why you're sicker than your mom was. You understand? Genetics plays a role. Okay, the differences in mercury concentration between rodents infected and not infected with intestinal helminths was not statistically significant. So I believe they're talking about the same rodent. We went over that one. This suggests, I'm sorry, I need to skip ahead. A. flavicolous body tissues, so the mice, had significantly lower mean average mercury concentrations than Myodes glariolus, than the voles. Okay, so 0.32 to 0.279. Assuming they were not infected by parasites. What happens when they're infected by parasites? If the animals were infected by intestinal parasites, the difference between both groups was insignificant. Because they're getting the toxic zinc in there. The voles had significantly lower mercury concentrations in liver and kidney tissues, the males, than females. What do we say about men and women detoxing around here? Ladies, you detox slower. Guys, you detox faster. This is not anything to rub into anybody, but it's just, ladies, you got to be patient. You guys store more toxicity. Why do you think the toxicity industry like cosmetics and perfume go after you? Because you can't get rid of it as well. And you are the bottleneck on fertility. If they can make you guys infertile, nothing happens. It doesn't matter if you have fertile men. If they can make the ladies infertile, nothing happens. You understand? What do you think birth control pills are there to do? Okay. Experimental studies on the lead accumulation in the cestode H. diminuta and its final host, Rattus norvegicus. I'm assuming that's a rat. I'm pretty sure that's what it said before. Yeah. It recently became clear that acanthocephalans, parasitizing mammals. So remember, we saw those acantho studies in fish. Do they infect mammals too? Yes. Can bioconcentrate, not just bioaccumulate, but bioconcentrate several heavy metals to conspicuously, very obvious, higher concentrations than the tissues of their definitive hosts. As cestodes are more abundant in terrestrial mammals, land mammals basically, than acantho, and thus potentially more useful in attempts toward passive as well as active biomonitoring, a very common tapeworm and its synanthropic mammalian hosts were selected for the present study. The tapeworm H. diminuta and experimentally infected male Wistar rats of the CDM strain were investigated with respect to their lead accumulation. The worms were allowed to grow for up to 5 weeks post-infection, followed by a 5 weeks oral lead exposure of the rats. After the exposure period, the rats were killed and the metal levels were determined in muscle, liver, intestine, testes, and kidneys of the rats, as well as the parasites. Lead concentrations were found to be 17 times higher in, I believe that this cestodes, they mean that was the tapeworm. Yeah. In the tapeworm than in the kidney, whereas metal levels in all other host tissues was below the detection limit. Thus, this study reveals that lead accumulation also occurs in cestodes, tapeworms, parasitizing mammals. They feed on it. So the other ones didn't accumulate. Maybe tapeworms like lead. So wouldn't that be crazy if they did this research on people and then let's say you found you had a tapeworm and then you could go, that means I have lead toxicity. So then you could deal with the lead toxicity. Wouldn't that be great? Do you think they're going to do that research for us? No. Effect of accumulation of heavy metals in the red fox intestine on the prevalence of its intestinal parasites. So could, as we're going to go into in this study, could a certain toxic metal kill parasites? Wait, toxins can kill parasites? No way. No way that toxins could kill parasites. So we can, like, just like you might be more sensitive to some toxin in your environment than your buddy might be more sensitive to some toxin in your environment than you. Could a certain strain of parasites be more sensitive to a certain toxic metal? But do you really want to try to kill a parasite with a toxic metal? So let's go. The intestines of 34 hunter-killed foxes were dissected to detect the occurrence of parasites. In 15 intestinal samples, parasitic intestinal helminths were found. Why don't they all have parasites? Why don't they all have parasites, Miss Hulda Clark? Why don't they all have parasites? They live in the same forest. They're eating the same foods. They're mating. Like, seriously. Many studies have demonstrated higher levels of heavy metals found in parasites rather than in the hosts. Some researchers report objective findings on bioremediation, or of cleaning up the biological system, ability of parasites to relieve their hosts of the heavy metal burden. The above findings might lead to the following presumption. By deliberately infecting a host with a parasite, the host could recover from intoxication. Can you believe what they just proposed? They're going to detox you by giving you parasites. Probably not going to detox you, because what they're going to want to do is they put the parasites in you and then they just try to kill the parasites with toxic stuff. It's a hell of a theory though, right? Concentrations of lead, chromium, copper, zinc, manganese, and nickel in the parasite-free intestines did not differ significantly from levels of the above heavy metals in the intestines with detected parasites. Okay, so they're saying in foxes and this parasite, there wasn't big differences between ones that had parasites and didn't. However, concentration of cadmium in the intestines of foxes without parasites was higher than that found in the intestines of animals with parasites. The difference was statistically significant. 3 times higher cadmium in the intestines of foxes without parasites than were found in the intestines of the animals with parasites. So I'm just going to throw this out there. The authors are going to say cadmium kills parasites, or parasites cannot live in a high enough cadmium concentration. Is that possible? Sure. But what if somehow, even though they weren't figuring it out, the parasites were cleaning out the cadmium? See what I'm saying? What we know is that the cadmium was higher in those without parasites and lower in the ones with parasites. That's what we know. That's what they measured. The rest is theory. The concentrations of lead, chromium, copper, zinc, manganese, and nickel in parasites were higher than the concentrations of the above metals in the host intestines. And the difference between the above concentrations was statistically significant. So here they just said that there was more of these. I mean, 5 of these are toxic. The only one that we want is zinc. And we've noticed a trend of parasites accumulating toxic metals. For those of you who don't know, can you take too much zinc? Yes, you can. Stop taking stupid doses of zinc if you're not testing. 30 milligrams of zinc a day if you're not testing. If you decide to go higher than that, do not ever come back and blame me that you took too much if you are not testing. You decide you want to go be a rebel and take huge doses? FAFO. Not my fault. Not my problem. You want to get your zinc right? You test. We test. How important is zinc to us? We measure zinc in the blood and the hair. That's how important it is. Okay, so we're going to talk more about land animals. Similarly, parasites of the genus M, or Moniliformis, parasitizing in rat intestine, contain cadmium in concentration 119 times higher than the intestines themselves. Shuris et al. Also investigated the ability of heavy metals to accumulate in M. hirudinaceus in pigs and found that cadmium, copper, iron, magnesium, manganese, nickel, and lead concentrations in parasites were up to 160 times higher than the amounts of the above metals in host tissues. So yeah, it might absorb some magnesium. Okay, but look at the rest of this list. So are parasites causing health problems? Possibly over time, yes. Are parasites possibly helping to relieve health problems? Yes. Can both be true? Yes. Our measurements confirm the findings of other authors showing that the parasite tissues contain higher levels of lead, chromium, zinc, copper, manganese, and nickel than the tissues of their hosts. The bioaccumulation factor reflecting the ratio of heavy metal level between parasite and host tissues was always greater than 1. Parasites always had more toxic stuff in them than the tissues. The highest accumulation capacity of lead was found in Mesocystoides species, which accumulated 21 times more lead than the fox's small intestine. Moreover, parasites can also accumulate other metals like chromium and manganese well. So 2 to 4.5 times higher, manganese 2 to 9 times higher. In accordance with other works, it was verified that red foxes with parasites had lower cadmium concentrations in their intestines than red foxes without parasites. As the concentration of cadmium in host intestines increases, prevalence of parasites decreases exponentially. The results of this study show that parasites may demonstrate higher sensitivity to the toxic effects of cadmium than their hosts do. Well, that parasite. Along with their limited ability to accumulate cadmium, higher sensitivity of parasites to its toxic effects can reduce probability of parasitization when a certain level of cadmium contamination in the host is reached. So it's like, well, and then let's just finish this up. The host whose concentration of cadmium in the intestines exceeded 0.05 milligrams per kilogram did not contain parasites in their intestines. So we get there's a difference between parasitized foxes and non-parasitized foxes. The idea of potentially making yourself toxic with cadmium to kill off one type of parasite is lunacy, right? But can parasites be affected by toxins? Yes. It's called ivermectin, hydroxychloroquine. Those toxins, black walnut, artemisia. Yeah, Let's keep going. Research note, relative concentrations of heavy metals in the parasites Ascaris sum, nematoda, and Fasciola hepatica, Digenia, and their respective porcine pigs. So Ascaris is in pigs, Digenia is in cows, definitive hosts. The concentrations of lead and cadmium, were significantly higher in the liver and kidney, respectively, of pigs than in their intestinal nematode parasites. Okay, so they're saying it was higher in the liver and kidney of pigs than their intestinal parasites. Okay, that's not following the pattern. There was no clear pattern in the distribution of lead within the ascarids, the pig parasite, but cadmium concentrations were highest in the intestine. Pigs don't detox well. They really don't. Don't eat pork. A parallel investigation of cattle naturally infected with the liver fluke, Fasciola hepatica, revealed interesting differences. Although the cadmium content, where was I? Although the cadmium content of F. hepatica was considerably lower than that in the tissues of cattle, the concentration of lead in the Digenian was on average 172 or 53 and 115 times higher than the muscle, kidney, and liver. Oh, so 172 times higher than the muscle, 53 times higher than the kidney, and 115 times higher than the liver. So, kidneys love cadmium. Cadmium and the kidneys go together. So, doesn't mean you don't accumulate in your liver. It just means cadmium preferentially gets accumulated in the kidneys. Furthermore, there was a significant positive correlation between the weight of individual F. hepatica and their lead burden. The liver fluke got bigger the more lead there was. Positive correlation. As the weight of the liver fluke went up, it went up with the lead burden. Do you think the parasite caused more lead, or do you think that more lead fed the parasite? I'm going to go with the latter. However, although lead concentrations in the liver fluke F. hepatica were considerably elevated above host tissue levels, the degree of heavy metal accumulation was relatively low when compared to that of acantho and cestodes of fish. So again, if we figured out that, oh, liver flukes, they get bigger and they flourish more with more lead, you think they're going to tell people with liver flukes that? Because then the people are going to go, why did I get lead poisoned? Where's the lead poisoning coming from? Let's fix that. They don't want you to know. This is why we have to go with animal studies. Okay. Visceral leishmaniasis is another parasite infection, and arsenic, an ancient poison contributing to antimonial. They're giving people antimony, a toxic metal, treatment failure in the Indian subcontinent. So they're giving people a toxic metal to treat a parasite infection, and too much arsenic is getting in the way. Have I told you that you can use one poison to treat another poison? Or you can use a toxin to treat a toxicity, a different toxicity. We talk about this like all the time. Antiparasitics are toxic to treat what is the result of another toxicity. Okay, antimony and arsenic are elements that have a long history of use as poisons, therapeutic agents, or cosmetics. When does something turn from a poison to a therapeutic agent? And are cosmetics basically just poisoning yourself through your skin? Pretty much. So this is all saying it's the same thing. Antimony and arsenic are toxic. And they say if we give you a little bit and we shut down your detox, then we might see a condition improve for a bit. And then that condition might come back later, or you might have a different condition show up later. Since the 1970s, millions of inhabitants of Asia have been chronically exposed to naturally occurring arsenic on a daily basis through the large-scale insertion of multiple shallow tube wells, which were originally installed for the provision of clean and safe drinking water. So they were trying to give them clean and safe drinking water, but they are poisoning the population. Whoopsie. Antimony and arsenic are metalloids belonging to group 15 of the periodic table. They're in the same group of the periodic table. That means they can replace each other in the tissues. That's what that means. That share many structural and chemical properties. They're like keys. They can fit into the same lock. It doesn't mean they do the same thing when they go into the lock. It means they can fit into the same lock. Only one of them is going to fit, though. Antimony resistance in leishmania parasites can be induced experimentally by exposure to stepwise increasing concentrations of sublethal concentrations of trivalent arsenite in culture. So imagine this. You have this parasite. It's happy to feed on arsenic. If it gets too much antimony, it might die. But it's going to absorb one of the two of them. So if you pre-fill the parasite with arsenic, you know how I said only one key is going to go into a lock? You can't get the antimony into the lock. You have used one toxin to prevent the effect of another toxin. Arsenite is arsenic, if you didn't know. If an individual who is chronically exposed to arsenic is infected with leishmania, the parasites would be exposed to arsenic due to its presence within organs of the lympho-reticular system. What is another food that is highly consumed in India that can often be full of arsenic? Is it rice? So is it just the water? Maybe not. Visceral leishmaniasis was almost eliminated in India in the 1950s and 60s as a secondary consequence of the National Malaria Eradication Program. Wait, so they were trying to get rid of one parasite and almost got rid of another parasite. What would be connecting the two parasites? Maybe that they worked on. Toxicity maybe. Maybe they both loved the same toxin. Or they were killing this toxin spreader. After the program's termination, the sandfly vector population increased, resulting in a resurgence of visceral leishmaniasis reaching epidemic proportions in the late 1970s. Primary unresponsiveness to antimony poisoning that they were giving them became evident during this epidemic when physicians found the prior standard 6-day antimonial regime to be grossly inadequate. It stopped working. If you don't know, the longer you give, like duration paradox says, the longer you give the poison, the less it's going to work and the more it's going to cause the original problem you were trying to fix with it. So they were trying to use antimony to undo the leishmaniasis problem, but the people kept getting more and more poisoned with arsenic, and then all of a sudden the people are now poisoned with arsenic and the antimony poison doesn't work. As a result, a 20-day regime at 10 milligrams per kilogram was introduced, which was further increased in subsequent years up to 20 milligrams per kilogram for 30 days to try to reduce the increasingly high rate of relapse. Wait, the parasites keep coming back? Oh, because you didn't deal with the toxicity problem. Despite this, cure rates with antimonial treatment continued to fall. The Indian subcontinent is the only place worldwide where endemic, as in it occurs there, it's a normal thing there, visceral leishmaniasis and significant arsenic contamination of the groundwater coexist. And it is from Bihar and Nepal that the highest rates of antimonial treatment failures are found. It's probably where they have the highest arsenic. Hey, what if sulfur comes into this? Gosh, what does sulfur do? Slows your detox. Both antimony and arsenic in their molecular form as antimonite and arsenite react with sulfhydryl groups and are thought to be leishmanicidal through depletion interference with the parasite's unique thiol system, which is dependent on the dithiol trypanothione. This system is responsible for maintaining the parasite's redox balance and thus its ability to defend against oxidative stress. In both laboratory and clinical resistant parasite isolates, an increase in protective thiols and overexpression of associated enzymes has been reported. Sulfur is toxic. Did we just go over how the foxes who had more cadmium in their intestines had less parasites? So can a toxin kill a parasite? Yes. Is sulfur toxic? Yes. Are they saying here that sulfur could reduce these parasites? This toxin reacts with sulfur. This toxin reacts with sulfur. and maybe it makes them just toxic enough to kill leishmaniasis. So let's keep going. Oh, now we get into Anthony Mawson. Anthony Mawson is the man who both Grant Genereux and I look up to greatly. He wrote a bunch of papers on vitamin A toxicity causing all sorts of problems, mainly through being released from the liver. His impression was that the liver cells would get damaged and bust open and spill out their contents, and it would be free retinol, free retinol esters, and free retinoic acid. Nobody ever talks about retinaldehyde, but retinaldehyde would be there too. So his theory was that the liver would just spill out a bunch of vitamin A, toxic vitamin A, into the system through damaging the liver cells. He did mention bile and cholestasis. I took it further, and I think it's really just cholestasis, but cholestasis includes the very cells that make the bile being busted open or leaking bile into the system. So it all kind of fits together. But Grant Genereux came up with the vitamin A toxicity idea, which works. Anthony Mawson has written 34 papers connecting vitamin A to various diseases because his son died of a brain tumor, and Anthony Mawson went and looked into it, and he was like, wow, vitamin A is totally related to brain tumors. Okay. So he was a man on a mission because one of his family members died from what he believed was vitamin A toxicity. Now, just so you know, when you see these papers, back in the day, he still believed that vitamin A was a vitamin. And to get your papers published, you kind of had to toe the line. But let's just go into this. So what do we talk about viruses as being? Viruses are toxicity. That's it. Whatever you think is a virus disease is a toxicity state. So, wow, what will we connect malaria, a parasite, a virus, a toxicity state, and the pathogenesis, the disease creation of Burkitt's lymphoma, a type of cancer? Pay attention to this first line. A geographical and causal connection has long been recognized between malaria, Epstein-Barr virus infection, and Burkitt's lymphoma, but the underlying mechanisms remain obscure. Modern medicine hasn't figured out what's causing them all at the same time. Why do they tend to come together in location? What can happen in location? Well, people eat the same things. They're exposed to the same toxins. They do the same things. They have the same factories polluting everything. Like, there's lots of reasons why it could be. But how is parasite, virus, and cancer connected? Potential clues are that the malaria parasite, Plasmodium falciparum, selectively absorbs vitamin A from the host. The malaria parasite loves vitamin A, and depends on it for its biological activities. Secondly, alterations in vitamin A metabolism have been implicated in many forms of cancer, including Burkitt's lymphoma. Alterations in vitamin A metabolism typically means slowed down ALDH. So you're accumulating retinaldehyde. The first author, Mawson, has proposed that the Merozoite stage malaria parasite emerging from the liver uses its absorbed vitamin A as a cell membrane destabilizer to invade the red blood cells, causing anemia, destroying the red blood cells, and other signs and symptoms of the disease as manifestations of an endogenous, released from within the body, form of hypervitaminosis A. Repeated episodes of malaria would therefore be expected to expose the tissues of affected individuals to potentially toxic doses of vitamin A. It is proposed that such episodes activate latent EBV infection. So dumping vitamin A, does it activate Epstein-Barr infection, latent Epstein-Barr infection? Or is it just a toxicity state that they call a virus? There is a naturopathic doctor here in my city who, after I talked to a couple of people who went to her and went to me, this woman in town, I mean, she's trying to help people, but she is hell-bent on diagnosing everyone with Epstein-Barr. Like, she has decided that everyone has Epstein-Barr. Okay, I could look at that and go, Well, yeah, she's looking in the wrong place. But if vitamin A toxicity is an epidemic, and vitamin A toxicity causes what we call Epstein-Barr, is she right in a way? She's looking for Epstein-Barr in everybody. She's looking for vitamin A toxicity in everybody. And she's finding it. If she looks long enough, she finds it. So thus, an endogenous form of retinoid as in vitamin A toxicity related to malaria infection may be the common factor linking frequent malaria, Epstein-Barr virus infection, and Burkitt's lymphoma, whereby prolonged exposure of lymphatic tissues, your body's sewer system, to high concentrations of retinoids, vitamin A, may combine to induce B-cell translocation and increase the risk of Burkitt's lymphoma. Okay, one connection of vitamin A to parasites. This is a theory paper. You think they're going to pay for people to study that? The pathogenesis of malaria, a new perspective. Now, this paper came out before the other one. And again, Dr. Mawson. We all owe him a great debt. A possible clue to such an understanding is that the malaria parasite P. falciparum selectively absorbs vitamin A from the host and appears to use it for its metabolism. Serum vitamin A levels are also reduced in children with malaria. Where does malaria tend to happen? Does it tend to happen in affluent, well-nourished countries? Or does it happen in countries where people are living in poverty and poor sanitation generally? It's the latter. So here was Anthony saying, although vitamin A is essential, no, it's not. But anyway, higher concentrations of vitamin A are cytotoxic, toxic to the cells, and pro-oxidant, so they cause oxidative damage. And potentially toxic quantities of the vitamin are stored in the liver. Yes. During their life cycle in the host, the parasites remain in the liver for several days before invading the red blood cells. The hypothesis proposed is that the parasites emerge from the liver packed with vitamin A, because that's where we store it, and use retinoic acid, the main biologically active metabolite of vitamin A, as a cell membrane destabilizer to invade the red blood cells throughout the body. I'm going to stop for a second. Cell membrane destabilizer. What is retinoic acid used for in aesthetic cosmetic medicine? It's called a yellow peel. It's a chemical peel. It dissolves layers, the top several layers of the cells on your face. So is retinoic acid a cell membrane destabilizer? Yes, it dissolves the freaking cells. Maybe you should, ladies, you should stop putting vitamin A on your face. The characteristic hemolysis, this is red blood cells busting open, and anemia of malaria. Well, that's how you get the anemia, is the red blood cells are bursting open. And other symptoms of the disease may thus be manifestations of an endogenous form of vitamin A intoxication released from the parasites. May also affect the fetus and cause preterm birth and fetal growth restriction as a function of the membranolytic, as in breaking up the membranes, and growth inhibitory effects of these compounds respectively. Oh, they're saying here, so it may be a manifestation of an endogenous form of vitamin A intoxication associated with high concentrations of retinoic acid, but low concentrations of retinol. So what are they doing? They're measuring the retinol in people. They only measure serum retinol. And then they look, oh, all these poor children have low serum retinol in these poverty-stricken countries. They need more vitamin A. No, they need more meat and they need more zinc. I've shown this multiple times. But could their retinol be transforming into other forms of vitamin A that they don't measure, like retinaldehyde and retinoic acid? They don't even usually measure retinoic acid, but they hardly ever measure retinaldehyde. Gosh, why would they avoid testing that one if that's the real problem? It's like they don't want to find it. Okay. Novel retinoid or vitamin A binding proteins from filarial parasites. The present study deals with the discovery and partial characterization of specific binding proteins for retinol and retinoic acid from filarial parasites, worms of the superfamily Filarioidea. So if you wanted to get rid of parasites, if we were to say that parasites like vitamin A, another toxin, and we remove that from their food supply, what is the easiest way to win a battle? Like, why did they do sieges in the Middle Ages? They just cut off all the food and all the other supplies to the castle. They just had to cut off the roads. And then the castle would eventually fall because they would starve, they would get dehydrated, they would get sick. All they had to do was stop the food. So how can we kill the parasites? Oh, we stop feeding them toxic metals. We stop feeding them vitamin A. And they slowly die and they pass. And what do we see people saying? As somebody just said, Fernando Espin in his super chat, I just saw this, so I wanted to read it. I passed a bunch of white specks for days, then a mother load of white noodle looking stuff. I was weak for 10 days. Was I just marinating in metals or did I crack my liver open? I slowed detox in C. I don't know if you mean in vitamin C, but so we didn't try. So Fernando's in the Love Your Liver program. We did not try, at least as far as I know, hopefully, Fernando, you didn't try to kill those parasites. They just died off on their own. Okay. But you stopped feeding them. This is what we're talking about. You starve the parasites, they die off. It's the easiest battle you can fight. And Fernando, I can't say that. He was a testimonial previously, but I don't want to say which one. So anyway, getting rid of these parasites can be done by simply starving them. You don't have to kill them. You don't poison yourself to kill the parasites. Don't do it. Fix the root cause, and then you get healthier and the parasites go away and they don't need to be there anymore. So, oh yeah, including those from 2 species of Onchocerca. That is the river blindness parasite. This is what they're trying to sell you on ivermectin so good for parasites because of the river blindness studies that they've done. What do we talk about here that vitamin A toxicity causes problems with? Is it vitamin A toxicity causes problems with the eyes? Parasite retinol binding protein and retinoic acid binding protein exhibited specificity for binding retinol and retinoic acid respectively. So the parasites have both of these. Pay attention. However, the levels of parasite retinol binding protein were between 4.8 and 5.8 picomoles per milligram, which is considerably higher than the corresponding levels of cellular retinol binding protein of mammals, and avian birds' origin. Wow, those parasites must really like vitamin A. Right? They must really like toxic things that they can accumulate. Now, is retinol and retinol binding protein probably going to be toxic to the parasite too? Yeah, which is what retinol binding protein protects them from. And cellular retinoic acid binding protein protects them from. Vitamin A is toxic to everything. Just because plants make it doesn't mean they love it. The funny thing is a lot of the fruits that people will try to say are the highest in nutrients are the same fruits that are the highest in vitamin A. What if the plant needs nutrients and antidotes to protect itself from the poison it makes? Right? It's like a knife. If you carry a knife, right, then you're carrying the knife because you might want to be able to protect yourself, which would mean hurting somebody else or threatening somebody else. But could that knife be used on you? Yes, it's just a weapon. So if vitamin A is toxic, it's going to be toxic to the plant too. And if we assumed that nutrients or vitamins are often antidotes for poisons, then it makes sense why the highest vitamin A foods would have the highest, what people think are nutrients. It's the highest antidotes. Okay. So next one. Retinoic acid signaling in gastrointestinal parasite infections. Lessons from mouse models. I think I need to find this with GLASP. Yep. Interestingly, vitamin A deficiency, which doesn't exist, and parasitic infections in human populations have extremely similar geographical distributions, with the same individuals experiencing both conditions. However, it is difficult to determine whether the relationship is causal or causative, as both are prevalent in areas of poor sanitation and poverty. which means they are not eating meat, which means they are deficient in zinc, which means they can't detox well. You need protein to detox. If you don't know this, your liver needs protein to run detox. So if you don't have zinc and you don't have enough protein to run detox, your detox system sucks. And what have we seen in the research? That if you give people zinc, if you give these poverty people meat or zinc or calories, their vitamin A levels go up, even though they're not taking in vitamin A. So where does that mean the vitamin A was stored? In their tissues, like their body fat and their liver. They were still getting plenty of vitamin A. You can correct vitamin A deficiency in the research with more protein, more muscle meat, more zinc, and sometimes even just more calories. And none of it has vitamin A in it. So where did the vitamin A come from? It came out of the liver and the body fat. I didn't read the title of this one. No, I did. Okay. So let's find the next one. Very few studies have investigated the effects of vitamin A on host resistance to gastrointestinal nematode infections in animal models, despite delivery of vitamin A to humans infected with intestinal parasites. So I want you to, are they trying to poison us while they don't do research on these topics? So there's not even animal studies on the effects of giving vitamin A to people infected with parasites. Yet, it is regularly done to give vitamin A to people with parasites. But there's very little research. What is Bill Gates' favorite vitamin? It's vitamin A. What does Bill Gates also believe is a very big problem? Parasites. Weird. What a weird coincidence. And you can go look up Bill Gates at the Parasite Museum. Bill Gates is part of the push for everybody has parasites. And Bill Gates loves vitamin A. And they give vitamin A to people with parasites, even though there's not even good animal research. The lack of investigations is also surprising given the importance of dietary absorption of vitamin A through the niche. The niche of these parasites. The importance of dietary absorption of vitamin A through the niche of these parasites, the GI tract. Wait. So what do most of the studies look at? When they look at vitamin A and parasites, they look at the immune system and they're measuring things in cells. Cell studies suck. So where are we missing the research? Any discussion concerning the supplementation of the diet of animal models of infection with vitamin A ligands, things that bind to the vitamin A receptor. So it's not just vitamin A that does that, if you didn't know. Should also include effects on the parasite rather than just on host immunity. They keep studying the immune system against parasites, but they're not actually studying the parasites. What are they avoiding? Some intestinal parasites are able to secrete retinol-binding proteins that can sequester or grab retinoic acid close to the parasite's niche. Gosh, they must like it. Studies have shown that the filarial parasite O. volvulus secretes a protein, this is the river blindness one, named OVFAR1 into the tissue surrounding it, which has high affinity binding sites for retinol and fatty acids, and therefore may contribute to the river blindness caused by this parasite through a reduced availability of retinol in the eye. How do we have Grant going 10 years on a low vitamin A diet and his eyes are just fine? I just went and I did a shooting competition the other day. I set a personal best. I'm 6.5 years in on this. Why are my eyes not falling out of my head if we need vitamin A? It's not, your eyes do not need any vitamin A. Okay. Or at least even if you only ate meat, from what Mike Roberto's research showed, if you do use the tiniest bit of vitamin A, you can get plenty from meat. So even if you ate an all meat diet, and if we still needed vitamin A, you could never get deficient. Other investigations have revealed that Brugia malii is able to take a radio-labeled retinoic acid. Oh, wait, another parasite likes vitamin A. Further, the retinoic acid was localized in high concentrations at early and late embryonic stages, suggesting a role for retinoic acid in its growth and development. So the parasite needs vitamin A for its growth and development. Parasites come about and thrive and feed on toxicity. Detailed analysis of the ABA1 allergen of the pig nematode Ascaris has also demonstrated retinol and retinoic acid binding capabilities. We can suspect that this sequestration or grabbing onto and holding onto of retinoic acid may be an immunomodulatory mechanism used by the parasite to locally deplete retinoic acid from the host and therefore alter the local immune response, possibly by decreasing Th2 responses and thus favoring its own survival, although this is yet to be concerned. They're trying to say that the parasites are like these evil things that are going to protect themselves so you don't kill them. Okay, what if you just stop feeding it? Interestingly, some parasites such as S. mansonii have been shown to have RXR-like receptors, retinoid X-like receptors, which seem to be important for female gene expression. Therefore, it would be interesting to assess whether retinoic acid can affect worm viability or fecundity, that's fertility, especially given the link to female gene expression. Currently, it is unknown whether gastrointestinal nematodes produce retinol-binding proteins or are able to respond to retinoic acid. Although genomic sequencing of these parasites will hopefully help to answer these questions. Why are they not studying this? Oh, here we go. Again, therefore, it is perhaps surprising that so little work has been carried out in this field, especially given the link between the prevalence of vitamin A insufficiency and parasitic infections in humans. We've shown that vitamin A deficiency is mainly a meat deficiency. I've shown in another Twitter thread, I believe, vitamin D deficiency is heavily related to meat deficiency. Of course, you need sun, but not having enough zinc and not having enough protein will give you a vitamin D deficiency in the research. Okay, we showed beriberi, thiamine deficiency is actually a meat deficiency. We keep showing all these diseases or just lab diseases are meat deficiency. Low vitamin A comes about because of not enough protein, not enough zinc. Oh, and I could probably show you, I'm not going to do it now. You could get blindness from a zinc deficiency. And maybe parasites really don't like zinc very much, and that would help them not stick around so much. Zinc also helps you detox vitamin A, cadmium, lead, copper, all these toxins. Oh, wait, mefloquine and psychosis. Mefloquine use, psychosis, and violence, a retinoid toxicity hypothesis. Again, Anthony Mawson. Mefloquine, what's that related to? Hydroxychloroquine. Recognize that? Hydroxychloroquine, mefloquine. Psychosis and violence. You start in so what I'm going to tell you before you start this, mefloquine and hydroxychloroquine and all the oquin drugs, they inhibit ALDH, your most important detox enzyme, which means they're going to build up retinaldehyde in you. Again, do they study retinaldehyde? No. Mefloquine use has been linked to severe gastrointestinal and neuropsychiatric adverse effects, including cognitive disturbances, anxiety, depression, psychosis, and violence. We don't have any of those today, do we? And that now there's so many antipsychotics being prescribed that there's commercials on TV for drugs to counter tardive dyskinesia, which is a common side effect of antipsychotic drugs. So they're not really advertising the antipsychotic drugs on TV. They're advertising the medicines that people can use for their side effects. So I'd say psychosis is going up too. The adverse effects of the drug are thought to result from the secondary consequences of hepatocellular injury, liver injury. In fact, mefloquine is known to cause a transient, that means temporary, anecteric, that's like jaundice, chemical hepatitis, liver inflammation. However, the mechanism of mefloquine-associated liver damage and the associated neuropsychiatric and behavioral effects of the drug are not well understood. Here we are. Where were we? There we are, right here. Mefloquine and other 8-aminoquinolines are the only antimalarial drugs that target the liver stage malarial parasites, which selectively absorb vitamin A from the host. Vitamin A is also stored mainly in the liver. In the research, stored in the liver in potentially poisonous concentrations. These observations suggest that both the therapeutic effectiveness of mefloquine and its adverse effects are related to the ability of the 8-aminoquinolines to alter the metabolism of retinoids, vitamin A and its relatives, the other retinoids. Alter the metabolism. I'm going to tell you what that means. It means slow down detox. Several lines of evidence support the hypothesis that mefloquine neurotoxicity and other adverse effects reflect an endogenous, from within the body, form of hypervitaminosis A due to a process involving mefloquine-induced aldehyde dehydrogenase inhibition. They're slowing detox, which causes the accumulation of retinoids in the liver, which then causes retinoid-induced hepatocellular damage, vitamin A-induced liver damage, the spillage of stored retinoids into the circulation, and the transport of these compounds to the gut and brain in toxic concentrations. That's basically toxic bile paradigm in a nutshell. It's just, I'm saying it's mostly happening via the bile, not necessarily liver cells bursting open. Is that part of it? Sure. But there's other ways for it to get into your bloodstream. But another vitamin A connection to malaria, a parasite. Anthony Mawson again, onchocerciasis associated morbidity. So, like damaging people, hypothesis. So this is an even older paper. Tissue concentrations of vitamin A in Onchocerca volvulus are about 8 times higher than that of the host. Parasites accumulate toxins. About 100,000 microfilariae die every day in heavily infected persons. That's not even people taking drugs to kill them. Onchocerciasis-associated morbidity may be due in part to the release of retinoids, vitamin A, from dying microfilariae, dying MFers, and their gradual accumulation to toxic concentrations in affected tissues. We're almost done. Again, malaria, but wait, there's more. A Plasmodium falciparum, malaria parasite, copper binding membrane protein with copper transport motifs. Motifs, it has such beautiful motifs. So wait, if malaria loved copper, what happens if we inhibit its ability to get copper? The copper chelator neocuproene inhibits malaria parasite ring to trophozoite transition in vitro, indicating a copper requirement for malaria parasite development. Y'all ever heard me say toxins of a feather flock together? So now we have malaria loves copper, malaria loves vitamin A, all these other parasites love vitamin A, and then they love toxic metals. Plasmodium falciparum parasites express a native protein with copper transporter characteristics that binds copper in vitro. It wants it. And that's already done. So let me wrap this up before I get into the super chats. So no more super chats. It's already noon. I still got to get to the inner circle. Parasites accumulate toxicity. They are a little toxicity depot or a toxicity sink, or even might say a toxicity filter. Okay. If you have parasites, in a certain way, they are helping you be less sick. They may also be causing problems. Can it be both? Yes. Is the solution to the parasites to nuke them? No. Does nuking them get rid of the underlying toxicity problem that you have? No. Does nuking them potentially release tons of toxicity in your system and damage your liver at the same time? You remember that mefloquine study? Yes. Toxins damage your liver. Damaging your liver slows detox. You start accumulating more toxicity if you slow detox. And then what happens if you nuked all the parasites, but still live in the same area. Are you probably going to get exposed to the same parasites again? Yeah. Are they going to find a hospitable area to live in? Yes. So we don't fix a toxicity problem, which is parasites, with more toxicity, even if it's just for the short term. We have people regularly expel parasites without trying to kill them, because what are we doing? We are starving them. Does a person who is starving fight back well? No. We're doing the most caring thing for your body that we can. We are reducing the toxicity. We are starving the parasites because they don't have the food and they don't want to be around anymore. They're like, dang, this place sucks. So then they leave and you poop them out and you see them in your poop. I had a period of time, actually, I didn't add this at the start. There was a period of time several years ago now, I mean, maybe 4 years ago, where in my poop for a while, as Fernando mentioned, I saw little white specks. Many of you have probably seen this. If you have, here's what I want you guys to do. If you're watching this later, or if you're watching it now, if you have passed what you think are parasites by doing the Love Your Liver program, please comment in the live chat and or comment in the comments below so people can see that this works. We've already had several people here now. I'm one of them. So I had those white specks in my poop for a while. And you know what happened at one point? They just stopped. And I was eating the same diet. So I didn't change my diet. And all of a sudden I'm seeing different little white specks of food in my poop. The little white specks were there for a while and then they were gone. Do you remember my story about the lady doing the niacin enemas? Parasites came out for a while and then they were gone. This is a common story. So stop supporting. And again, parasites and cancer. The parasites don't cause the cancer. The parasites, in a way, are protecting you from the cancer for as long as they can until you overspill the parasites. The parasites can't hold anymore, and then you get cancer. Were the parasites there first? Yes, but you got to understand the order. There's an order to this. Okay, so I think that wraps it up. I mean, you can do whatever you want. But if you're continuing, what I hear a big story of in the alternative community is people say, I have to keep treating parasites. I had a family member who was like, I have parasites and I have to keep treating them. Another family member said they had giardia. It keeps coming back. But the person says, if I eat well, it goes away. Are you seeing the patterns? We fix all sorts of gut issues that people will say are connected to parasites, yet we don't treat parasites. So if you're out there doing the Love Your Liver program and you're trying to treat parasites, you're doing it wrong. Do the program better and stop poisoning your liver at the same time you're trying to love your liver. Don't do that. So let's get into the super chats. Let me go back to this. Okay. Hound Jog, thank you for the super chat, says, hello, doc. If the bird's nest was parasites, do you need to worry about the eggs? Okay. I'm just going to do a weird little analogy here. So like if you had a baby, newborn baby needs milk, and you took away its food source, the mother, what's going to happen to the baby? It's not going to live. You're not providing the environment, the food, calories, protein necessary for it to grow. So do you need to worry about like this? This is Hound Jog. This is good. You're still kind of stuck in the old thinking. No, you don't have to go after the eggs. Because if you actually literally killed the adult parasites, like just imagine if all the adults on the planet disappeared, are the children going to survive? No, they're not going to survive. They're not equipped for it, and there wouldn't be food around for them, and they don't know how to make their own food. At least most of them don't. So, no, you don't have to worry about eggs. This is all just like, this is why they're trying to treat people with like different herbs and different drugs. You got to use this drug or this herb for the eggs, and you got to use this one for the larva, and you got to use this one for the full-grown one. No. You starve them all by not taking in toxins and then giving your body the nutrients that it needs to protect itself and to detox the toxins. That's the same thing. We use the same concepts and principles for everything. Okay, so that's that one. So thank you very much for that. LeBrad Pass, thank you for the super chat, says, hi doc, what are good baseline blood tests you can get? 8 months into Love Your Liver, 34-year-old male. Okay, well, let me say this first, because there's a lot of people out there who are getting hair tests and blood tests. Maybe they hear about what we use and they get a hair test and a blood test and they get what we use. If you hand somebody who's never played a trumpet before and they have no idea what they're doing, are they going to make music? No. It is the poor musician who blames their instrument. What I'm getting at is you can do all the testing you want, but if you do not know how to interpret it, you're not going to know what to do, and you could make yourself worse. There are people out there, even people within the Love Your Liver program, who are ordering tests, and I don't teach people in the Love Your Liver program how to interpret your tests. And they are doing the wrong things with them because they are, let me say it at the very start, giant ASS assuming that just being able to take the work I've done for nearly 20 years, and I've been doing hair testing since 2013, and try to think that you could just look at labs and analyze them with no training and no context and none of that stuff. And even worse, if you go read Dr. Lawrence Wilson's stuff on the internet, you're dead wrong. If you're following anybody who does his work, you're effing yourself up. I save people who said they were dying from doing that stuff. I helped save them. One woman almost lost her marriage after 3 years because she was going insane. They just kept telling her to eat more and more vegetables. That was their advice. You're just detoxing. You're just retracing. Your whole life is falling apart and has been steadily for 3 years. Your husband's about ready to leave you. You're just detoxing. Just have more vegetables. Nutritional balancing is garbage. But that's what you're going to find the most of, the information on the internet. Sometimes the shittiest advice is the loudest, which is really what that is. So, okay, to give you an idea of how much care I take in this and how much context you need. My recent seminar, I forget when it was, maybe was it February or January? I don't remember. I did an online seminar. It was over 2.5 days. It was 18 hours long. 18 hours long. And I gave the people who came to it, we're doing what I call a seminar study group, which is about 2 to 3 hours session, one session of 2 to 3 hours a month for a year. I included in the price of it a 2 to 3 hour session once a month for a year. Do I want people to understand this? Yeah. And did all the our health facilitators, even though they might have done it before, they still came to it to catch up on new things they might have missed or things they might not have heard before. And then I made the seminar available to people as something they could purchase later or that they could watch again. People can do really dumb things with good information. So, with that said, if I give you these blood tests and I give you enough noose, enough rope to hang yourself with, that's not my fault. I just warned you. We use a hair test from Trace Elements. You can actually order your own hair test from from our website, from our store. You can order just a hair test. If you order just the hair test, you are ordering just the hair test. You are not ordering evaluation of it. You're ordering just a hair test. That's it. What does just a hair test mean? It means just the hair test results. It does not mean you're buying any interpretation. So email Julie or Sarah saying, Dr. Smith said I could order a hair test and get an interpretation of it. No, if you buy the hair test, you're buying just the hair test. Then there is something on the store called Just the Essentials, the do-it-yourself program. You can order the hair test, the 4 blood tests that I want, and I think it's about 8 hours of me going over what these results mean and what you might choose to do with this information. Again, what you might choose to do with this interpretation based on the information I give you when you're looking at your own test. And I'm going to give you a lot of if this, then this, but if this, then this. Watch out for this, as you guys all know that I do in the whole program. It is never, we are not as mindless as saying, like, people on Twitter. I saw one guy who was like, everybody should be taking 100 milligrams of zinc a day. And I was like, oh, the creatine people now, 45 grams of creatine a day. I made a joke on there the other day. When is the creatine boofing going to start? If you don't know what boofing is, that's putting drugs up your, or drugs or alcohol or whatever up your rear end to get it to absorb faster. Like that, it was a joke, but yeah, somebody's going to be boofing creatine sooner or later because they just can't take all that orally, whatever. It's just so stupid. All the mega dosing you're seeing is like psyop. And it's also because that stuff doesn't work. And if they want to use it to suppress your detox, they have to go to higher doses. So, okay. Let's see. So, oh, no, the blood test. Let me just hit on that. So the just the essentials do-it-yourself test. It's a shorter version of it's the prep videos. If you get a testing and consultation package with any of our health facilitators, Nathan or Hope or Kelsey, if you get a package, you would get the hair test and the 4 blood tests, an hour-long consultation with the facilitator to review it with you, to go over what they suggest you do. And then you get access to the office hours, and myself and Kelsey and Hope and Nathan all do 1 session a week of office hours. So there are 4 separate sessions that you could pick to go to. You could go to all of them. You could go to none of them. If you wanted 4 times, if you wanted help to ask questions 4 times a week, you could go to every single office hours. That's included in everything. So if you buy a package, you get access to my office hours. Everybody gets access to all the office hours. Okay. So just think, if you sign up with Kelsey, you can go to Kelsey's office hours. You can go to mine. You can go to Hope's. You can go to Nathan's. You can go to any of them. You could go to all of them for 6 months. And you get lifetime access to the Love Your Liver program as part of that too. So that's that. Now the 4 blood tests that I look at: serum retinol or blood vitamin A, copper, serum copper. I like plasma zinc. If you can only get serum zinc, that's okay, but I like plasma better, and serum ferritin, or it's just called ferritin. Those are the 4 blood tests I use to assess toxicity and to treat deficiencies. Now, let me just give you a common example. People get their tests back, and let's say they're looking at zinc and copper. And on their blood test, their zinc is low. And on the hair test, their zinc is high, or vice versa. Their zinc is high on the hair, or zinc is high on the blood tests, and it's low on their hair test. And then they start going, what do I do? And that's why I take my job so seriously. Why do we measure both of them? So we can see that. So we're not in the dark like the people who think only blood tests tell you what you need to know, or the people who say only hair tests tell you what you need to know. And then I'm the only person who is integrating the 2 of them. Again, you can mess yourself up with these tests. So if you choose to do it, if you want to do it on your own, I would highly suggest you get the Just the Essentials DIY program if you want to save a little money and try to do it yourself. A lot of times people who buy the Just the Essentials DIY program, they get it, they get the information, they get the tests, and they go, oh shoot, I can't handle doing this. I don't trust myself to do the answers. Like, do I do this much or do I do this much? If you're one of those indecisive types, don't do the Just the Essentials type. You need help. And if you want to just buy the hair test, it's just a hair test. Hair testing is the most nuanced and context dependent testing out there. People who think hair testing analysis, the analysis of the hair test is simple and straightforward, have no idea what they're talking about. No idea. Absolutely no idea. People who have taken my course understand this now. And then when you see the hair test change over time, what people are taught in the hair test field is they look at just one hair test as like, this is an encapsulation of you at this moment. No, it's actually showing an average of the time that hair growth happened, of what was moving up and down in the blood. And then they think that it's good for every single mineral. There's all sorts of contaminations that can happen. You have hard water, your hair calcium might be high. You bleach your hair, your calcium might be high. You take this hair sample from the wrong end of long hair, the calcium will be high. You use Head and Shoulders once in 6 months, the zinc might be high. There's all sorts of things that can happen that can screw up the hair test because it's outside of your body. So we have to know what to look for. First, you have to know what to look for if the hair test looks weird. Because that's the one drawback, is it's outside of your body. But yeah, that whole like dandruff shampoo once in 6 months, if you have a long hair sample, it can absolutely show up. Absolutely contaminate your hair sample for up to 6 months. And not even knowing that can be a problem. So all I'm saying is I take my job very seriously, and I'm going to let you know how seriously I take it. And that if you want to FAFO on doing it on your own, I mean, that's what you're doing. Hope's been doing the testing. Hope did my first seminar, or was it my second? Yeah, I don't think Hope did the first seminar. Hope did the second seminar, and she's come to all of them since. Kelsey came to my last one. I think Nathan came to 2 ago. So they've all been working on it. They all caught on very quickly. That's why they're here. But they will all tell you that no, it's not just something you just like. I would ask them, Hope and Kelsey and Nathan, to say, if I gave you these tests before you ever took the seminar from me, and you did what you thought you should do then versus what you would do with this now with what you know, would that be drastically different? And I'm sure all of them would say absolutely yes. So, okay. Next one. Oh yeah. So I already did Fernando's super chat. Beth said, thank you for the super chat, Beth. She said, oh, and thank you for the super chat, Fernando. Beth said, I would love if you could do an episode and or a King Hero interview on how psychiatric drugs mess people up and how to best come off of them. #tragic. This is, I don't know if I can do that. Partially, it opens me up to a whole lot of liability. Partially, it's people who are on psychiatric meds. So those are messed. They were already messed up, and now they're on psychiatric meds, which are messing them up. And their decision-making skills could be quite poor. And usually, by the time people are that far into the medical system, they don't totally buy into what I do. It's like a lot of people call themselves alternative, right? Until they get cancer or they think they have parasites. And then they're like, oh my gosh, we got to do pharma. I'm like, you were like a debutante. You like feigned allegiance to the alternative side. But then when shit got real, you just run to big pharma. Pick an approach. Stick with it. So, That's a tough one. I might be able to do something on that. However, still, how people, I mean, I had, as an example, this was something that happened in my history. I had a woman come to me who she was actually an she went to high school with my ex-wife. And she came and she was extremely anxious, and she was on, I think if I recall correctly, it was 1 milligram of Klonopin like 3 times a day. That's a hefty dose from what I recall. This was years ago. This maybe it was like started 10 years ago. Okay. She read stuff on the internet, and the people on the internet told her, do not take anything with your weaning or it will screw you up. She was so anxious and she believed the internet more than me. And she said, I'm not going to take anything else. And I'm sitting there, I'm like, you won't even try like topical magnesium or some potassium just to help bring the anxiety down? No. I'm just going to wean. I'm just going to wean off the Klonopin. And I said, okay. So I did some research for her, and I found, okay, with benzos, anti-anxiety meds, one of the first things you want to do for weaning is switch from the strong ones that have very short half-lives, which is why people have to dose them 3 times a day, because they spike and then they drop, and then they spike and then they drop. And they give, the more you spike, the more when the drop comes, the more withdrawal you get. So the advice on the internet was like, take somebody who's on Klonopin and switch them to Valium. Okay, benzodiazepam, I believe. Switch them. Then they gave equivalents. I think it's 10 milligrams of Valium to 1 milligram of Klonopin, if I recall correctly. And they said this way the come downs will be gentler. The withdrawals, when they're hitting the between times, it's easier. And it's easier to drop over time. I asked this woman, Matej, thank you very much. Matej, right? I said it right the second time, right? Matej, thank you very much. She said, to the best doc ever. I appreciate that. But so this woman would not switch. She was like, nope, I'm not going to switch the med. Not going to change anything. So I'm sitting there going, okay, so you won't take anything to help, and you won't let me do the advice that I found from many people who have successfully weaned off before and switch you to a less potent but longer acting benzo. Okay. There's a book out there called Let Them by Mel Robbins. I can't change how people think and how they feel. That's why those of you who think that I'm going to go on social media and like pursue people and try to convince them, no, I don't do that. I am not here to chase people. I am here to present my information, and the people who are ready for it and who are willing to listen and whose brain is in the right place will grok it, and they will stay. I am not here to waste my time and energy convincing other people. So let her decide what she wants to do. I said, okay, fine. You don't want to do any of that stuff? I never brought it up again. And it took, at 5 years of weaning, 5 years of her contacting my office and telling us whether we could drop the dose or not that month. The pharmacy, the compounding pharmacy made special pills of a certain weight just for her for every month for 5 years. She was down to, so she was on 1 milligram 3 times a day. 3 times a day, 1 milligram. I believe that was the dose. By the end, she was down to like 0.0106 milligrams a day. So she was down to 1/100th of her original dose. And she was so anxious, she wanted to do like smaller and smaller cuts. And she was already down 99%. 99%. And she still wouldn't jump off. So dealing with people who are on psychiatric meds, you're dealing with a psychiatric issue. And a lot of these people are, if somebody went to pharmaceuticals in the beginning for depression or whatever, if that was their first go-to, they may not be the type that's going to grok this work quick enough to, because every time something bad happens, they're going to run right back to pharmaceuticals. That's what tends to happen until they're finally sick of it enough that they're going to suffer. And let me explain what happens with these psychiatric meds. There's a specific paper on Prozac showing that Prozac specifically inhibits vitamin A detox or vitamin A metabolism. Let me find it just real quick. What is Prozac again? What's the word? Fluoxetine. I'll share my screen, don't worry. There it is. I want you to understand what SSRIs and all these other psych meds are doing. Read this title: Direct Inhibition of Retinoic Acid Catabolism by Fluoxetine. Direct detox slowing down of your vitamin A detox pathways. That's what it's doing. So let's just say retinoic acid in your brain was connected to depression. Okay. So what would medicine do to that? They say, well, you got too much retinoic acid. Let's slow it down. You can build up retinaldehyde. Well, that would be kind of funny if I think retinaldehyde is one of the bigger problems, and people who go on antidepressants then start gaining a bunch of weight. Because one of the things that retinaldehyde can do is retinaldehyde can go backwards through ADH and be turned into retinol and then be turned into retinol esters and stored in the body fat. ADH, alcohol dehydrogenase, is reversible. ALDH is not. So, let me get the link. Okay. So I want you to understand what happens when people try to come off of these psychiatric meds that are specifically slowing down vitamin A detox. What they're doing is all of a sudden they're taking the brakes off of their detox system. They start processing. Now they've built up vitamin A for however many months or years they've been on this drug. When you think of withdrawals, the only thing you should think of is you stopped taking something that was slowing down your detox. And then your detox speeds up again and you're dumping tons of toxins into your system. And that is why you feel bad. Why would your body need a poison? That makes no sense. But if we know poisons slow detox, and then you take your foot off the brake and you start detoxing stuff, but after you stop the poison, you're even more toxic than when you began, which is why you started probably taking the drug or the poison. And now you have it rushing into your system. And also, as your detox speeds up, you make more bile than you used to, but now your bile is more toxic and you leak it more, and then you feel bad. And you either get to ride that out, or you get to maybe learn about toxic bile paradigm and the Love Your Liver program and the stuff we do here to make it easier. Does that mean it's going to be easy? No. Sometimes people are cold turkey people. Sometimes people are weaning people. I don't know which one you are. So you kind of got to pick. It can be pretty rough to go cold turkey, but then usually things are over faster. The people who are kind of more sensitive or more scared of the slam that happens when you cold turkey, then they get to wean. But then also, like that other lady, you can get stuck worried about ever being off of it. Okay, no more super chats, please. That's this. But so LeBrad, about those lab tests, if you want to order any other labs, like CBC, CMP, TSH, those are all showing symptoms. So you can see some of your symptoms, like CBC, complete blood count, shows anemia, let's say. But does that tell you how to fix it? No. Oh, you're welcome, Beth. Okay, so let's hit on. We had the one, Joseph. Thank you for the super chat. It was how to poop more. I want to have. This is the last super chat. So if you post any more super chats, they're going on next week. Okay. So how to poop more. Well, I'm going to hit on the simple things here. So you have to decide which of these things you're already doing, or that you know. You have to think about, okay, so as an example, soluble fiber. If you don't understand toxic bile paradigm or toxic bile theory yet, Joseph, you're going to want to go watch episodes 71 and 53 so you understand what's going on with bile and your poop. Bile triggers your poop. More bile equals more pooping. However, if the bile's going the wrong way, if you stimulate bile and it's going the wrong way into your blood, then you feel worse. So typically with constipation, you're either not making very much bile. If it was going into your, so bile is the trigger to poop. Bile is the main trigger to poop. I'm going to say it again. Bile in your intestines is your main trigger to poop. Not enough bile in your intestines means you're constipated, or not enough bile touching your intestines. Let me clarify that. Too much bile touching your intestines gives you diarrhea. So you want to be a normal person with a normal amount of bile that's not toxic. That's your goal. But people who are here are not necessarily there yet, right? That's why we're all trying to get better. Okay. So with constipation, either you're not making very much bile that's going in your intestines, or the bile is going back into your bloodstream from the liver itself. And then you're just getting you just feel bad, like hypothyroid, which has constipation. You're getting hypothyroid symptoms, as an example, from the bile going back into your bloodstream, and it's not helping you poop. So let me go over the things that can help you poop. First of all, as one guy said, I'm just throwing these things out there. Each person is going to have to look at these things and understand, does that apply to me? Do I want to test it? Okay. I remember reading this one powerlifting coach, and he was talking about if you're trying to gain weight and you're not crapping 3 times a day, he said very simply, you're not eating enough. Okay, so first of all, if you want to poop a lot, you have to eat a lot. But if you eat a ton and you just get more constipated, then don't do that. Like all I'm saying is you have to have enough food in you to poop. So if you're under eating would also tend to lead to hypothyroid. If you're under eating, you're not going to poop well. So that's what we could say. We don't say you have to overeat, but if you're under eating, you're not going to poop well. There's not enough stuff to push. It's called the mass movement complex. Mass in your bowels, movement, right? So if you got the little rabbit poops, they're not going to move along very well because they're like this big, right? And your intestine can stretch out like how big? So it doesn't mean that it necessarily. You ever tried to contract your bicep when it's really small and you feel how weak you are at this angle? Like, so if your intestines are like this, they can't squeeze very well. So if there's not enough food, there's not going to be much poop. So you must not be under eating. And this can happen to people on this program because when you go to Whole Foods, meats and beans and grains and the right fruits and that kind of stuff, sometimes people they undereat calories because whole foods are very filling. And then they come to me going, why am I losing weight? And I'm like, are you eating enough food? Oh, wait, I checked. I'm not eating enough food. There you go. Okay, so next one. Water. Water lubricates your bowels. So if you are not drinking enough water, your poop will be effectively drier and it won't move very well. Not to use an analogy of lube, but if you don't have enough water down there in a certain way, you don't have enough lube for the poop to move, okay? So if you're constantly peeing like more yellowish, you might not be drinking enough water. And if you're constipated and you're having more yellowish pee all the time, then maybe you need more water. If you're peeing clear all the time or really light yellow, you're probably getting too many fluids. Okay, you want yellow pee. Yellow. Everybody knows, if you picture the color of pee in your mind, we think of it as yellow, right? We don't think of pee. We don't think of a clear glass of water. We also don't think of like stinky brown stuff. That's dehydration. And then clear or really light yellow is not enough fluids. So get the right amount of fluids. People say my hydration video in the Love Your Liver program is one of some people have said that video was worth the whole program. It's not very hard to get your hydration right, but you got to know the principles, like the soup analogy as I go over. So that's just simple stuff. Then you can get into things like soluble fiber. Now, soluble fiber does two things. Soluble fiber does stimulate bile dumping. It does stimulate bile getting pumped out. Depends on where it's going though, right? What if it goes right back into your blood? You can still have the trigger happen, but the bile go the wrong way. And if you don't make enough bile, let's say you tend towards constipation because you're not putting enough bile in the intestines for whatever reason. Soluble fiber triggers bile dumping. Soluble fiber adsorbs bile in it. So it takes it away from touching the intestines. You got to understand that. Charcoal constipates people a lot because charcoal's super good at adsorbing, grabbing onto bile. The bile then in the charcoal is not touching the intestines anymore. So you're not triggering pooping if you take too much. You take too much soluble. So think of it as the early phase of soluble fiber in your body. You dump bile. Okay. If you take more soluble fiber than the bile you dump, the soluble fiber soaks it all up and it's not there to trigger your pooping. This is where people make the mistake of soluble fiber all the time, or like the carnivores where they're like, fiber's terrible for your guts. And you have the vegans going, fiber's great for your guts. And it's like, it can be both. It's where the bile is going. This is where toxic bile paradigm answers constipation and diarrhea. So you have to figure out the right amount of soluble fiber for your digestive system at this time. If you were like, wow, I got on the Love Your Liver program, doc, and I started eating more fiber and I got more constipated, stop. Eat less. It's not helping you. Or some people come on, they're on the Love Your Liver program. I started eating more soluble fiber. Man, I poop 3 times a day now like a champ. Great. You're doing a good thing. Doc, I ate way too much soluble fiber and I got diarrhea. Tone it back. Like, listen to your body. So just know soluble fiber can do both. Then there's sun fiber. Sun fiber has research in it. Sun fiber is really a food for your gut bacteria. And fixing your probiotics, your gut bacteria, is one of the quickest ways you can help yourself. Sun fiber is partially hydrolyzed guar gum. So it's a fiber that has been, if you want to call it digested, hydrolyzed into an easier to eat form for your gut bacteria. And it has research showing it can help constipation, and it has research showing it can help diarrhea. What did I just say soluble fiber could, cause or help? So think of sun fiber more as food for your gut bacteria. And just so you know, there is detailed instructions. Joe put it on the screen. Detailed instructions in a link on the back of the package in the how to use. And that link is also on the product page in our store. So I just had as an example, I just had a woman send us an email the other day. I got your sun fiber and I started taking a full scoop a day and I've got gut stuff going on now. And I sent her back. I said, here's detailed instructions on how to use it. It's linked on the package. It's also here. It's also on the product page. Please read it and then adjust what you're taking and start taking less. Joe, that's the product information. It's up towards the top. Right there. Go down. The link right there. That is the link. Organic sun fiber instructions. And it's like pages of how to start on it. We don't just say like 1 scoop of sun fiber for everybody. Some of you tried sun fiber and you're like, it's pretty potent stuff. And some people have to start slow and work their way up. Same thing with the lactoferrin. So sun fiber can help. Now, the big thing that helps a lot of people poop is oral magnesium. Magnesium you take by mouth, not topical. Topical helps your systemic levels of magnesium, like our magnesium lotion, which is amazing. Oral magnesium that you take helps you poop. Oh, Beth was saying, my friend recently solved long-term constipation with sun fiber. When you get the right thing, it helps. Oh, and if on the probiotic note, I'm just going to generally make a statement. If you're going to try a probiotic, don't try mixed probiotics first. If you're male, try Bifido. If you're female, try Lactobacillus. See if they help. If they make you feel worse, stop immediately. Start with a pinch. Open the pill, take a pinch. Start slow. When you change, so if all this talk we do about bile, your gut bacteria can make your bile more toxic or less toxic. This is why, for those of you who want to test this, go and find a popular probiotic on Amazon. Look at the 5-star reviews and look at the 1-star reviews. You'll find people who say the 5-star reviews, this probiotic changed my life, made everything so much better, fixed all my gut problems. And then you'll find people on the 1-star who are like, this probiotic gave me irritable bowel syndrome and I can't get rid of it. Don't start with a full pill. Start with a pinch. That's what I have people do with sun fiber. That's what I have people do with lactoferrin. Because you don't want to be one of those 1-star people on Amazon who just kept taking a pill a day for 2 weeks going, it's going to kick in, it's going to kick in, it's going to kick in, and now you got big problems. If a probiotic is going to help you, you will notice it very quickly. And if it's going to make you worse, you will notice it very quickly. Sun fiber feeds both lactobacillus and bifido. Still, we have people start slow on it. Okay, so oral magnesium. So when you read about magnesium online, the better absorbed a magnesium is, the less good it is at making you poop. Or what I mean is you have to take higher doses to get you to poop. So the more of it that's absorbed, the less it helps you poop. The magnesiums that are the most poorly absorbed are the ones that help you poop. What are those? Magnesium oxide, magnesium hydroxide, also known as milk of magnesia, magnesium citrate, like the natural vitality calm with no flavorings. These are the ones that help poop the most with the lowest doses. Okay. Magnesium. No, I wouldn't use magnesium carbonate. Don't do that. That's a buffer for like niacin and other stuff. Don't use magnesium carbonate as a quote unquote laxative. But if you're out there taking magnesium malate or magnesium glycinate, the ones that are absorbed better, you're going to have to take a lot more pills to make yourself poop if that's what you're trying to do. It'll cost you a lot more money. And none of the magnesiums work very well to raise magnesium on a hair test. Watch my topical magnesium video. Topical magnesium is how you raise your systemic levels, your cellular magnesium. Oral magnesium raises your pooping. And if you're taking magnesium and you're like, well, it doesn't help me poop, you didn't take enough. If you take enough magnesium, you will poop. So if you tell me I took 5 pills of magnesium, I still didn't poop, take more, you will poop. It's kind of a forceful thing, but the nice thing about magnesium is it's not habit forming. Like if you take magnesium to help you poop one day, you don't have to take it again the next day necessarily to help you poop unless you're chronically constipated. Okay. Next thing, potassium. Getting your potassium right. I have live streams on both topical magnesium and potassium. Potassium helps your thyroid. Potassium helps your energy. Potassium, by helping your energy and your thyroid, can help you poop. don't take potassium like a laxative. It's not like magnesium pills. You're not trying to take so much potassium that you crap, or you'll send yourself to the hospital eventually. Don't do that. You're taking potassium to help the whole system run better. Okay, low potassium will cause constipation. Okay, so there's that. Then there's, I don't mind if you find, and can you mix and match these things? Yes. Some people will poop better with insoluble fiber. Sometimes insoluble fiber helps people poop better. Okay. The last thing I believe is prunes or prune juice, which are dried plums or the juice of dried plums. Is this optimal? No. People are going to say, but isn't there vitamin A in it? Yes. Do I think that people pooping on a regular basis is more important than the little bit of vitamin A that they would have to use from the amount of prunes that would help them poop? I don't think it's that big of a deal. Also, if you don't take the prunes or the prune juice with fat, you won't absorb very much of the vitamin A from them. And you can help stimulate pooping with that. So I'm pretty sure I went over the main, what we call the Love Your Liver pooping helpers or constipation helpers. That's all in the order of adding stuff. So for those of you who are in the Love Your Liver program, in the program, look for the order of adding stuff. It's up towards the top. And I think these are listed there. There might be one I forgot. That's Joe just put up the link for the Love Your Liver program if you want to join. It is a lifetime membership, one fee for lifetime. And for those of you who were in it before, you have all been upgraded to a lifetime membership. So if you get an email saying you need to re-up your membership, look at the email. It will say it is free. It will say zero charge. $0 is the charge. So if you get that email and you don't read it and you go, oh God, I don't want to get charged again, you're not getting charged. Nobody else is getting charged once you're in it for the lifetime, which is from here on out. And if you were in it before, you're now in it for a lifetime for no extra payments. So don't get that email and freak out and cancel it if you don't want to, because you're not going to be charged again. It used to be a monthly thing, and we changed that. Okay. And so Houndjog had a good question. This will be the last thing I answer. Houndjog was saying, what if charcoal helps you poop? This is a weird phenomenon. This is rare. It happens. There are people who will paradoxically or react the opposite of expected to all sorts of things. My ex-wife taught me all about this. My ex-wife reacted the opposite to everything I gave her. So a lot of you ladies out there, especially, or you guys, both ladies and guys who are sensitive and who have the opposite reactions, you can all thank my ex-wife, because she's the one I learned about going slow and paradoxical reactors. And if you can't take it yet, then don't take it. My ex was the type where I'd say, why don't you try this? Try a small dose. Tell me how it's going. What would I get? She would take a full dose, and a week later, she'd tell me, I haven't slept in a week. And I'm like, why did you not tell me? Like, I told you to take a smaller dose, and I told you to tell me what was going on, and you didn't. She was absolutely my worst client ever in terms of listening. Absolutely the worst ever. But it taught me a ton. So you guys all benefited from her stubbornness, if you will. But charcoal, some people dump more bile with charcoal than it adsorbs, and they poop better. Some people, most people get constipated. Like, I can take 2 tablespoons of charcoal a day and poop fine. Most people are like, what? Like, I take a quarter teaspoon. I can only take a quarter teaspoon without it constipating me. That's like normal. Quarter teaspoon, half teaspoon. I'm taking 2 tablespoons a day. Sometimes I normally do 1 a day. Sometimes I'll do 2. I'm a good pooper. So if you are one of the ones who can take charcoal and it helps you poop, realize you are blessed. And that means you can take as much charcoal as you choose to. I would highly suggest you take as much as you can, short of getting diarrhea. Because if you, like I'm saying, if you take too much and you dump too much bile, you can get diarrhea because you dump more bile than the charcoal can soak up. Some people tell me they can't take charcoal because it induces a bile dump that they don't feel good with. And I'm like, well, I guess you can't take it. Okay. We listen to the body's signals. So what does it mean? It means you poop better when you take charcoal. It means that somehow the charcoal's triggering you to dump bile. That's it. That's all I know. With a lot of these mechanisms, like people want mechanisms, I'm like, I don't know. I want you guys to remember, I figured out most of this stuff through working with people, through working with myself, observing myself, testing out these things. And then once I felt comfortable with it, then I would do it with normal people and I would work with them and I would have them do it, start slow, watch your responses, then tell me what happened. The opposite of what my ex-wife would do. Tell me what happened so I can learn from this. So I can see that, like, okay, you want to know one thing I know about oral magnesium? This is my estimate. Like, right, I'm not doing research studies, but this is my estimate. And I seem to be pretty good at pattern recognition. About 1 in 20 people can actually get good magnesium levels from pills. About 1 in 20. The rest of them, to raise their magnesium levels, have to do topical magnesium. That's just the way it is. How do you know which one you are? If you're not testing and you don't know how to read a hair test, you don't know at all. If you feel better on magnesium, topical magnesium, it means you need it. If you feel different from taking oral magnesium, you're absorbing some of it. Is it enough? I had a guy come to me. He was taking magnesium glycinate, magnesium malate, and magnesium threonate. Rich guy, podcaster. I had to tell him when I got his hair test, oral magnesium doesn't work for you. Your magnesium sucks. But the threonate crosses the blood-brain barrier. If you need magnesium in your brain, and magnesium threonate is the only magnesium that's ever crossed the blood-brain barrier, you'd be dead. Do you understand? That supplement only came out like 20 years ago. And so if people needed magnesium in their brains before then, and they never got it, they would be dead. Magnesium threonate is an exercise in lying through science. All the studies that showed going into the brain and all that stuff, all sponsored by the company that makes it. Realize you are being lied to in the research every day at every minute. So, that is everything for today. I'm winding down. Got to go do the inner circle now. So, hope you all enjoyed that. Let's just run through the links, Joe. So, if you want to go to our main website, that is nutritiondetective.com. That is where you can find our health facilitators, Nathan and Hope and Kelsey. And you can find the testing and consultation packages. That's where you can find the Love Your Liver program under membership. Then you can find our supplements. If you are a Love Your Liver member, you get access to the VIP store and you can buy them at a discount. Please, if you're not a member of the Love Your Liver program, don't apply for the VIP store. You're not going to get in. Julie checks all the addresses and you're just wasting time. So if you want discounts on all the supplements we sell, which includes things that we don't have on the main store, you got to be a member of the Love Your Liver program. Then there's the Love Your Liver program. If you want to go straight to that, that's a do-it-yourself program. That is at members.nutritiondetective.com. It's a mini social network. It's a forum, if you want to call it that. That's where you can talk to other people doing this. And that's where you can find the program called the Love Your Liver program on the menu. If you go down and you see Love Your Liver program, that's where it is. But that is a do-it-yourself program along with all the other people in the community where you can ask questions and all that stuff. Then, you can find me here on YouTube at youtube.com/nutritiondetective. I have a Rumble channel. I don't know what the address is on that, but it's probably not hard to find. If you search for Nutrition Detective on there, I'm sure you'll find me. I'm on Twitter at NutriDetect, NutriDetect. And then, I wanted to explain one more thing. Hound Jog asked a question at the start. Okay. I'm off to go do my inner circle. What is the inner circle? The inner circle is a weekly, another Q&A session that I do right after this. It is inside the Love Your Liver program. It is not included in the Love Your Liver program membership. It's a separate monthly fee. Okay. Those people get to ask questions. They get to have me answering these questions in a more private setting than, let's say, YouTube here. So I can answer things in a more straightforward way and not have to hedge my wording. And then for people who want to see the inner circle, but don't necessarily want to ask questions, and they don't mind seeing it a month later, the Advanced Detox program has those videos a month after I record them. So you don't get to ask questions, but you can see it at a greatly reduced price. So those are only available inside the Love Your Liver program. Oh yeah, and then Joe linked my GLASP study. So if you wanted to look for a study that I talked about, you can go to that GLASP link and it'll show you the stuff I highlighted, and you can find the actual study and you can go find it again. So that has all of my GLASP stuff on it. So that is, I think that's it. I'm going to wrap it up. I got to go do the inner circle right now because I got to go pick up Milo at school. I got a time limit today. Okay. I talk so much, my voice is going, I think. Anyway, you all have a great day. I will see you all next time. And remember, don't try to kill the parasites. Starve them. Okay? Bye now.